স্যার কিছু একটা বল স্যার কিছু বলছেন? Now it's everything is clear
Some noises are coming.
till now. Noise, মানে
Okay, it's all right. Okay, thank you.
Good morning to all, and today we are in the second day of our refresher course. Today we have among us Dr. Nikhil Ranjan Jana. He is a more senior professor. He doesn't need any formal introduction, but we need to do it. Uh, he did his PhD in chemistry from Indian Institute of Technology, Kharagpur. And the most important thing that we need to know that his work, that is, his laboratory focuses on chemical synthesis of colloidal nanobioconjugate and their application as bioimagining nanoprobe and for prevention curing of neurodegenerative diseases. He has authored or co-authored about 225 peer-reviewed articles and 4 books on basic and applied science, with a primary focus on tuning of nanoparticle surface chemistry toward various biomedical applications. Sir, now I want you take from this part, start your lecture.
Thanks to the organizers for a quick introduction. I am now going to share full screen. Yeah. So is this okay now? Can you see the full screen?
No, sir, can't see.
Share screen. Yeah. Everyone see?
Yeah, now we can see.
Okay. Okay. So good morning to everybody. So today we will talk about the biomedical application of nanoparticles, and these are the 4 specific areas that I will try to cover. First one is in general the role of nanometer length scale in biology. Means why nanoscience is important in biology, and after that I will discuss a few methods of making nanoparticles and nanobioconjugates. That's mostly a chemistry aspect. And after that, I will discuss mostly on how we use these nanoparticles for different biomedical applications and various approaches of making nanobioconjugates and how we use them. So let's start with some introductions of nanoscience. You already know, most of you have heard about this. So it's a science at nanometer length scale, typically in the range of 1 to 100 nanometers. Not only it's a synthesis, it's application of these length scale or materials or device fabrications. And 1 nanometer is actually 10 to the power minus 9 meter. It's very difficult to imagine that size. And in order to have some ideas about this, I have shown in the bottom of the slide several images. And we have some idea about tennis ball size, size of a dot or spheroid. We can see in our eyes. We have some idea about bacteria, virus. They are very small. We can't see in our own eyes, but we can see under microscope. These are conventional optical microscope. Remember, optical microscopes are very common, but electron microscope, that's the modern days microscopes, there we can see much small things. Those are not commonly available in most of the common labs, and those are actually modern discoveries of electron microscopes. So the molecules we usually don't see, whatever we make as a chemist or even some biologists, they deal with biomolecules, we usually don't see them either our own eyes or even in conventional optical microscope. And nanoparticles are somewhere between molecules and smallest size of the things that we use to see our conventional light microscopes, roughly between 1 to 100 nanometers. The well-known example is coronavirus. This is about 50 nanometers. And a glucose molecule that we know, most common molecule, is about 1 nanometer. So you can roughly try to imagine that we are dealing with a nanoscience means actually somewhere the size between a glucose molecule and coronavirus, somewhere in this range. And wherever you see the nanoscience is actually interdisciplinary science. It's neither taught in physics, chemistry, or biology or any other subjects. It's because physics, chemistry, biology are the conventional subjects that we used to learn in school, college, universities. Those are developed long back, and nanoscience is a quite recent subject areas, which are actually interdisciplinary in a sense that you have to have experts and knowledges that covers physics, chemistry, biology, and some of the subjects that you can see here: applied physics, engineering science, material science, supramolecular chemistry, molecular biologies, biomolecules. Many of them are large size, like polymers. So, macromolecular biologies. These are all involved in nanoscience. And interdisciplinary, we are not aware of interdisciplinary nature of subject. We usually, from the school life, we we becomes an expert. Particularly after college, we becomes expert on certain subjects. But in nanoscience, you need to have expertise. Maybe in some subject you have to learn more, but you need to have overall expertise, general expertise on other subjects. Otherwise, it's almost impossible to learn nanoscience. History of nanoscience is actually history of gold nanoparticle synthesis and property studies. Michael Faraday is one of the pioneers. He has many different works, but he also synthesized colloidal gold nanoparticles, which are pink color. And he was interested to understand the interaction of light with such a small particles. That time, nano term was not common. Richard Zsigmondy was a German scientist who actually developed synthetic methods of making gold colloids, small size. And that time, electron microscope was not available, but he used some other scattering-based approaches to determine the size, which are actually very small, about 10 or like that. Langmuir film, actually it's a monolayers of molecules, which are actually two-dimensional nanosystems. Richard Feynman, you heard about his books, plenty of rooms at the bottom. Invention of electron microscope, 1930 to 50s, which becomes commercialized after 50s. Birth of cluster science, these are all linked to history of nanoscience. And this is the most important microscope, was electron microscope, where we use electron beam instead of light for capturing image of small particles, nanoparticles. And there are many other names of similar microscopes. Some of them are scanning electron microscope, that's mostly image the surface. But electron microscope is actually you can image even something inside below surface. And most important is that we can see nanomaterials using these microscopes. And seeing is believing. So we can have much more understanding. We can convince others that what we are making. This is another version of microscope, scanning tunneling microscope, where actually you can image molecules. So whatever molecules, which was kind of chemistry was kind of invisible science, that is no more true nowadays. And with these tools of electron microscopies, of various imaging tools, characterization tools, nanoscience becomes very, very interesting and important areas of science. One important aspect in nanoscience is the materials has size-dependent property. Here I have shown three materials: silver, gold, and cadmium selenide. You can see they have different colors. For example, silver, you can see wide range of color just by changing their size and shape. But these changes you have to do in nanometer scales, typically between 10 to 100 nanometers. Same for gold. They are colloidal particles. They have very strong colors. These colors can be tuned by changing their size or shape. The bottom one is cadmium selenide. It's quantum dot, semiconductor nanoparticles. And just by varying the size between 1 to 10 nanometers, 6 nanometers, between 1 to 6 nanometer, 1, 2, 3, 4, 5, 6. 6 nanometer, every nanometers change, you will get different emission color. These colors are emission. Colloidal solutions, if you shine light with UV light, you will get different colors depending on their size. So material property is size dependent. Same material, just by varying size or shape, you can get different colors. This is not possible by conventional chemical. If you want to make colors, different colors of an organic compound, you have to make different molecules, different bonding structures. But here, same material, just by adding size or shape, you get these colors, these properties. This is another interesting example. These quantum dots that I have shown earlier, they have, when you shine light in the colloidal solutions, the average property you don't see blinking. Fluorescence is stable, continuous. But when you go down to single particle imaging, you will see they blink like stars, blinking stars. What that indicates? We made a mistake by describing quantum dots average property. Average property of fluorescence is actually stable fluorescence, no blinking, no change in fluorescence intensity. But when you go down to single particle, you see these blinkings. The impact is that sometimes we make mistakes. If we think everything on the based on average properties, particularly in biology, enzymes, macromolecules work as a single molecule, and we have to understand their properties at single molecule level. This is another subject area, again Nobel Prize winning subject area, super resolution microscopy imaging, where we can break the diffraction limit of light. We can use light to image nanoparticles. You know, conventional light like visible light, 400 nanometer, you cannot see. Using this light, you cannot see something less than 200 nanometer things. But using super resolution approach, you can use the same lights microscopes, but with different techniques, we can image nanomaterials. So this is a type of super resolution microscopy where nanoparticles, nanoprobes, which has fluorescence blinking property, that can be used for imaging, super resolution imaging of nanoparticles. For example, here you see the amyloid fibrils, which are lysozyme fibrils. These fibrils are linked, these are protein fibrils, few nanometer diameters, micron size lengths. These are responsible to Alzheimer, Huntington's, many different neuro diseases. You can image these particles, these materials using super resolution microscopes. So these are some of the examples. The nanoscience, why this subject is important in terms of biology. Here you will see another aspects. There are many important biosystems which are nanometer length scales. Why nature use those? And in nanobiotechnology, what we try to do is we try to understand the functional role of nanoscale units in life processes. And separately, we have understanding of size-dependent property of materials. We try to bridge these two knowledges to design some new nanomaterials that can be used for unique biomedical applications. For example, wireless therapy, brain delivery, detecting molecules at single molecule level sensitivities, tracking molecular events, video imaging of molecular events, what's going on inside cells, and many other things. This is a wide range of examples where you can see biology, which kind of units, micro bubbles, microtubules, collecting vesicles, lipid raft. You might have, whoever in biology, they might have this coronavirus, and diatom microstructures, silica particles, silica porous structures, iron oxide. These are present in certain bacteria for navigating in the sea using Earth's magnetic field. Lotus microstructures, kidney stones, early stage kidney stone or any other. These stones are actually early stage of nucleus and they make this kind of particles. Bone microstructures, they are actually nanoplates of hydroxyapatite, calcium phosphate. Malaria pigment, these are actually iron chelate based materials. In a double helix, they are major minor groups which are actually of nano size. So many examples of nano structures in biology. Why nature? They have every nanosystems in biology, they have specific functions. And once we know those functions, we can better use similar kind of functions, or we can sometimes do much better these, and we can design nanomaterials for doing better these biofunctions. And if we want to do these biomedical applications, we have to know that there are size effects. Typically, we try to make particles about 100 or less than 5 nanometer to 100 nanometers in this range. And in this size range, there are many different size effects. Renal clearance. If you want something inject in your body and then it should be clear, then size must be less than 5-6 nanometers. If you want tumor targeting via enhanced permeation retention effect (EPR), then size should be 100, between 100 to 200 nanometers. There are many other. If you want magnetic nanoparticle based contrast agent (MRI), contrast agent size small is better. If you want cellular uptake of your materials, more efficient uptake, size should be between 10 to 50 nanometers. So many other issues. But you have to also know the colloidal science, colloidal property of materials, how you can disperse them better. You have to do some surface chemistry in order to have good water dispersibility, because in biological media is actually water, organic solvents. Here I am showing some examples of nanoparticles. You can see here they have names and scale bars are present. From those you can understand the nature of particles and quality of particles. They have different size, shape, and these are all electron microscopic images. Light microscope cannot be used to see them. And these are another example. Some of them are fluorescent, some of them are magnetic, and middle line. So these are all barium titanate, for example. This is sound responsive materials. You can use ultrasound to do some things, some chemistries on their surface. And nitrogen fluorine dot TiO2. You can use visible light, sunlight, for killing bacteria or virus using these particles, and so many others. Nano green tea. The green tea has some ingredient chemicals. You can convert them. These chemicals you can use for converting them into nano particle form of green tea and use for different applications here. Protein inhibiting protein aggregations which are linked to Alzheimer or Huntington's. So once we make these kind of particles, we have to design them in a way that how they will interact with biological interface. Biological interface means cell and molecules, biomolecules, for example, protein, DNA. These are most common things. So you have to design nanoparticle in a way that they can interact with cell, enter into cells, and they can be delivered into different subcellular compartments, then interact with some biomolecules, proteins present there. How can we make particles? You have to make particles in a way that they can interact as per our interest. And for that, you have to know many things. Here is an example. Let's say if you inject your nanoparticles into body, either via skin, via injection or inhalation or by oral delivery, where they will go. And middle one is actually composition. There are many ways of making different compositions. There is a size effect. Depending on size, if it is very small, they can get out of body, and depending on their size, they will take different time, few hours to few days get out. But in most cases, if it is between 10 to 100 nanometer, they don't get out. They stay in the body. They deposit mostly in lungs, brain, not brain, lungs, then heart, kidney. They are the most common. And if you have a tumor, they will deposit in the tumor. But they hardly go into the brains because there are extreme protection. Nothing can go easily into the brains. So, but they can go certain parts of your organ. Most common is kidney, lungs, heart. And if you want renal clearance, they have microstructures in our globular systems, glomerular filtration systems in our kidney. You can see the structures and microstructures, and ideally they should be less than 6 nanometers. Otherwise, they will deposit in the kidney. They will never go out of kidney. And ideally, if they are biodegradable, enzymatically degradable, then also there is a chance that even in a long time they can become small and excrete from the bodies. And if you want cellular entry, again there are barriers. First barrier is membrane, cell membrane. They are lipophilic membranes made of phospholipids, a kind of compact liquid crystal-like structures. And also they have many different proteins, they can sense. Depending on that structure, they will sense something. If they want something good, they will in, they will pass. They will it will enter into the body by endocytosis. There are many types of endocytosis: lipid wrap, clathrin mediated endocytosis, caveolin mediated endocytosis. And once they enter into cell, there is again various biochemical processing mechanisms. Depending on that, they might be enzymatically digested by most cases. Some cases they will go into certain targets. Also, there is a blood-brain barrier. If you want to deliver something into brains, there is a compact endothelial cell layers. And in addition to that, there are astrocytes. Those layers they have to overcome. So you have to design nano drugs that can overcome those barriers if you want to deliver something into brain, into neuron cells in the brains. And there are a lot of issues designing nanomedicines for restoring our thinking process. We may or may not know that almost all of us have a issues of thinking process. This is more linked to social issues. Everybody have social issues, like we have problems of teaching our childs, problem of getting jobs, many social problems, personal family problems. Everybody have thinking problems, and these social problems are linked to our brain diseases. It's not just brain diseases have a major reasons coming from social issues, and we need to design nanomedicines that can partially solve these issues. There are tremendous research efforts in these directions, and major issue is bypassing the blood-brain barriers and delivering something into the brains. Now we will discuss very quickly about some of the nanoparticles that we make. Most widely used methods, most well-developed methods. How to control size. Nanoparticles, we have to make particles of defined size, 5 nanometer, 1 nanometer, 10 nanometer, 50 nanometers. For inorganic nanoparticles, for example, gold, silver, semiconductors, cadmium selenide, these are the standard methods. Nucleation growth. You start with the metal salt precursors, and then convert them into ion to metal atoms, then agglomerate those atoms into small clusters, then grow into particles, nanoparticles, then stop the growth. That's called nucleation growth mechanisms. And chemists are major players in controlling size, and without them it's almost impossible to make high quality particles. There are a lot of issues. How to control nucleation growth. And there are chemical reaction mechanisms which are too complex. That's linked to nucleation growth. Actually, you can see some problems here. There are certain nanoparticle, certain nucleation means the agglomeration of formation of small particles. They sometimes catalyze whole nucleation growth processes, and as a result, polydispersed particle size. Particles that forms at the early stage, they grow faster. So as a result, small and large particle mixtures. So this is an example. If you are a unsmart persons, or maybe you are very beginning in the research in nanoscience, or maybe you thought that textbook knowledge is enough to understand or make particles, you will end up with these kind of sizes. These are gold and silver particles. They are most known particles. A lot of papers on this. But if you start making these, you will end up with these particles. These are useless in most of the applications. And making small particle is easy. Because nucleation growth is a very complex process, you will always end up with mixture of small and large particles. It's better to use a very strong capping agent so that you stop the growth at the early stage, and you will end up with small particles between, let's say, 1 to 5 nanometers. But if you want more control, want to get size somewhere above 100 nanometer or 10 nanometers in good size, this is one of the approach, seeding growth. You start with a small seed, then grow in a good conditions without secondary nucleations. This is an example. You start with a 2-3 nanometer seed and grow into 5, 8, 17, 37 nanometers. Is one example. And another good approach is you start with a bad polydispersed particles and then use heat in presence of some capping agents or conditions, heating condition, temperature and time. You can end up with a monodispersed particles. But remember, this is very strict conditions and not applicable to every particles. But most of the commercially available gold, silver, quantum dot, iron oxide, those are actually developed by following these methods. You can make a relatively large scale. And now I am showing 3 different well-known, well-established methods we commonly use for making particles, gold. This is one example, gold, spherical gold particles. This was developed by Richard Zsigmondy long back. This is modified, and now it's established method. So basically, boiling gold salt, we are adding aqueous solution of gold salt under boiling condition. We are adding citrate, sodium citrate. Their citrate in boiling condition acts as reducing agent. It degrades and makes radicals. These radicals act as a reducing agent. And high temperature, citrate also acts as a capping agent. Citrate as reducing and capping agent. And high temperature, 100 degree and about 30 minutes, that actually ostral ripening. Initially, polydispersed size becomes monodispersed. And between roughly about 10 nanometers, but depending on capping agents, if you use small amount of citrate, then size can be increased. Typically, you can vary between 10 to 50 nanometers. Early stage versus final product. Final product, you can see very monodispersed. But their colloidal stability is very poor. So we have to again process further if you want to use for different applications. This is another famous method, burst method. We can make small particles 1 to 3 nanometer size gold capped with surfactant, thiol-based surfactant. These particles are, you can make large scale. So this is a 2-phase synthetic method. Forget about the details methods, but strong capping agent is used and strong reducing agent, sodium borohydride reducing agent. And once you make this, you heat it in presence of capping agents and in different solvent, different temperature and time. You will get to start with the small particles and end up with a different size between 5 to 10 nanometers. Very good quality. Ostral ripening. Gold nanorod synthesis. Start with a gold salt, make a small spherical seed and a growth condition in presence of these surfactants. You end up with the different shapes, rods of different lengths. Very standard methods. And quantum dots, 2 years back it got Nobel Prize. Cadmium selenide capped with zinc sulfide. Cadmium selenide fluorescence, you can tune the fluorescence by varying the size. This is a method, high temperature injection method. What's the temperature? About 300 degrees. Solvent is a high boiling organic solvent. It's called it's hydrocarbon or resin. It's a high boiling but liquid. So in high temperature you can do nucleus and growth. You are injecting. Why injection? Injecting precursor in a very short time, so that you can control nucleation and growth within very short time. You can stop the reaction, let's say 30 seconds, 60 seconds, 1 minute, 2 minutes, stop the reaction, and depending on reaction time you can get different particles, 1 nanometer, 2 nanometer, 5 nanometer. And these particles are fluorescent semiconductor. Once you synthesize this cadmium selenide, you have to sell. You have to have selling of zinc sulfide so that the fluorescence is stable. Iron oxide, the magnetic nanoparticles. Again, you start with a iron salt, then heat at 300 degrees, and during heating process, you generate iron oxide particles starting from your iron oleate or long chain fatty acid salt of iron, converted into iron oxides. Depending on the heating temperature, capping agents, you get different size. In some selected cases, you can make very narrow disc hollow particles. Hollow particles can be synthesized. Hollow particles have different colors. For example, gold, silvers. You start with a gold silver particle and then mix with silver. Sorry, you start with a silver particle and mix with gold salt. You will end up with a hollow gold particles. It has a different effect. It's called Kirkandal effect. What is that? This nano silver particles are very reactive, and in presence of gold salt, this is ligand, it's an exchange process. Gold salt is converted into gold atoms, reductions, and silver zero is converted into silver ion. And these reactions is very rapid and place exchange. Because of this place exchange, silver changes place, moves from inward to outwards. That leads to pore formations. Every silver particle is converted into hollow gold particles. This can be adapted on many different hollow particle synthesis. So I have just quickly showed you high quality nanoparticle synthesis. Gold, silver, they have colors, plasmods. Because of that, they can be used for different optical detection applications. For example, just by colors, you can naked eye detection of different DNA, RNA, proteins, and also you can use surface and Raman spectroscopy based detection of molecular fingerprints using gold silvers. Quantum dots, they have fluorescence, that's how you can use them for bioimaging, fluorescence imaging based applications. Magnetic nanoparticles you can again use for MRI or magnetic separation. Okay, these are examples, roadmaps. Once you make particles, for biomedical application, you need to do a lot of surface processes, surface chemistries. Because just nanoparticles, they won't interact with your biological systems. In many cases, because they are insoluble in water. Many cases, they interact with their surface molecules, whatever present in the surface. Through these surface molecules, they interact with cells or biological molecules. And that interface you have to design by chemical approaches. Once you design, you can do various applications, for example, imaging probes, delivery carrier, and so forth. Now I will show some examples. So everything has to be colloidal and water dispersible. They should have terminated with some chemicals, biochemicals, so that cells or biomolecule can read and understand what is there. That's how they interact. How to make them? Before that, you have to know what's the type of nanoparticles, what's the surface. These are the types of surface. But we like to make this kind of particles. Nanoparticles with cross-linked cells, so that they are very stable. You can do organic chemistry at the surface. So as synthesized nanoparticles are converted into colloidal, this kind of core cell type particles, porous or cross-linked cells by silica coating, polymer coatings, kind of things. Once you make these particles, they are very low volume. You can precipitate them, redisperse them by varying the medium, by varying the conditions, by adding salt, removing salt. So these are the types of particles that these are the examples of our labs. We make these particles. Nanoparticles terminated with different biomolecules, galactose, biotin, folate, trihaloids, TPP, triphenyl phosphonium, phenylalanides, some kind of peptides. And also we need to control their numbers per particles, because these are kind of valencies. Because through these biomolecules they interact with biological interface, and these are kind of valencies. By controlling this valency, you can control the interaction. And this control, so by controlling this, we can control their uptake with cells. This is a cell uptake. We can These are the particles that we make, and depending on the particle size, surface chemistry, there are different uptake mechanisms. And different uptake means endocytic or non-endocytic uptake. And these uptakes can control their processings, like processing intracellular processing. They may be trapped or they may be free. And so depending on design, all this depends on design, size. So if they are trapped, they are not accessible for subcellular target. If they are not trapped, they are accessible. But again, where they will target, that depends on their chemistry, surface chemistry.
So these are important.
So what are the applications? Imaging probes, small animal imaging, biosensors. Once we have particles, functional particles, in most cases we culture cells, or in biological interface, we put our colloidal solution, water dispersible particles, into the culture media, and during the Brownian motions, they, depending on the surface, they interact with cells. And after washing and processing things, we have to measure by microscopy, fluorescence, or any other microscopies. Ideally, we need to label different parts of cells. If they are fluorescent, quantum dots, for example, very powerful materials, we need to label, we need to design quantum dot based probes with different surface chemistry, so that they can label different parts of cells. Why quantum dots? Why not conventional probes? Because they bleach. This is an example. Quantum dots label in the nucleus, cell nucleus, and outsides are labeled with conventional organic dyes. And you can see within second, if you continuously expose with light, within few seconds, within minute, the organic dyes completely bleach, but quantum dot don't bleach. That's the advantage of nanoprobes. Quantum dot or gold silver particles, they never bleach. Fluorescence can be used for video imaging, tracking single particles, single molecules. So this is a one design. This is a quantum dot with some peptides, different kind of peptides. This one is very interesting, that it can be used for imaging tumors or cells that are cancer cells that are in advanced stage. So that in advanced stage, they need enzymes, MMP, matrix metalloprotein, that can cleave what is that called, interface this cellular, what is that called, interface, cell and matrix interface. So matrix can be cleaved by this MMP. And so that cells can invade different parts of your body. They can travel from tumor to different sides of your body, and they can invade different parts, and tumor spread everywhere in the body. So at this advanced stage, they use MMP, some enzymes. These quantum dots are designed in a way that MMP can cleave, then convert the nanoparticle from anionic to cationic. And they can cationic particle can label your cells, cancer cells, and tumors. That's how they can label tumors that are in advanced stage. You can use nanocarriers that can be used to deliver proteins or small molecules into nucleus. Cell nucleus, many cases you have to deliver drugs into nucleus, therapeutic proteins into nucleus. And for that, you need to design a carriers. These are some of the examples. And once you deliver drugs into nucleus, they are more effective. They are more effective in therapeutic performance. You also need to deliver drugs into mitochondria. This is an example how you design carriers and load your small molecule drugs in carriers and then deliver into mitochondria for more efficient therapies. This is another example where this is a conventional TiO2 nanoparticle, but it is doped with nitrogen and fluorine so that it captures sunlight, visible light. And once you spray into your fruit and then expose with your light, visible light or sunlight, it can kill fungus. Here it is shown that tomato can be where the fungus is there and it's exposed with visible light, fungus are killed in presence of sunlight. Because these nanoparticles can capture visible light and generate reactive oxygen species. I told that neuron cells or neurodegenerative diseases that are involved in brains, we have to deliver materials into brains. And these are list of small molecules. We know that they can inhibit protein aggregations linked to those diseases. Once you convert these small molecules into their nanoparticle forms, these nanoparticles are more effectively delivered into those neuron cells and into brains, and they can be much more effective for. So these are kind of nano drugs. So molecular drugs are converted into nano drugs for more effective brain delivery and neuron therapies. These are the model systems that we use. These are the protein fibrils that are linked to diseases, and we use in the cell models and mouse models to show the performance of designed nanodrugs. These are examples. They are protein fibrils. The wire-like structures are fibrils, are small dots, are actually our designed nanodrugs. They bind to these fibrils, and if they bind at the early stage, they stop these protein fibrillations. They stop the growth of fibrillations. That's how they inhibit protein aggregations. And in cell models, you can see they bind. These are the examples. These are the cells, and the fluorescent dots are actually the aggregated proteins that are responsible to diseases. They are tagged with GFP. And if we treat these cells with our nanodrugs, don't see that those dots. These protein fibrillations are inhibited. Protein aggregations are inhibited. These are more fine structures, protein estimations to check whether the aggregated proteins are degraded into small particles or not. It's a nano green tea. How do we design? How we use them for inhibiting protein aggregations in cell and mouse models. This is a nano tree halogens. These are the small molecules, tree halogens, that are converted into nano forms and then use them to treat in cell models and mouse models. They are in brains. These are brain systems, so that they are in the brains, these nano drugs in the brains because of these trials, and they inhibit these Huntington aggregations and inhibit those aggregation processes. Once these proteins aggregates are there, they are inside cells, you need to need a drugs that can remove those drugs from those cells. And we use an autophagy-induced nanoparticles. So nanoparticles are designed in a way that they induce protein induce this autophagy inside cells, and this autophagy process is actually clears protein aggregates. Autophagy is a cell cleaning process. So once our nanodrugs are there into cells, inside cells, they attach with aggregated proteins and remove those proteins from cells via autophagies. We can also design nanodrugs that are sound responsive. We can use ultrasound for various therapeutic purpose. This is a therapeutic ultrasound. Ultrasound that don't destroy our normal cells, don't destroy our cells, but if we have drugs, nano drugs that are sound responsive, they can do some therapies without disturbing normal cells. These are the types of nanoparticles. They are piezoelectric nanoparticles. Piezoelectric means they generate charges in presence of pressure. These are nanorods, barium titanate. These are hydroxyapatite nanorods, nanowires. More anisotropic, more piezoelectric. These are piezoelectric constant values. More piezoelectric means they generate more charges. More charges mean more electrochemistry can be possible at the surface. Electrochemical reactions, piezocatalysis. And we can also convert these particles into their films, so that if cells are grown on those films, we can use sound-based therapy. These are the small molecules that can be excited by sound ultrasound. How? Ultrasound can generate luminescence called sonoluminescence. They are visible colors, blue, red, yellow colors, mostly blue colors. Those lights can be used for exciting these molecules. Not every molecule, certain molecules. And these excitations can lead to generation of reactive oxygen species. This is more principle of piezocatalysis. The piezoelectric nanoparticle, well-known example is barium titanate. Under pressure, these particles can generate charges, positive, negative, and these charges are responsible for generating reactive oxygen species via, if oxygen is present, they can capture electron and convert into superoxides, followed by hydroxyl, hydrogen peroxide. So every piezoelectric materials are centers for this piezocatalysis. And using these piezoelectric materials, we can use ultrasound-based therapy, cell therapy, tumor therapy, autophagy inductions, ultrasound-based, ultrasonic clearing of bacterial strains, ultrasound-based blood-brain barrier crossings. These are examples, real examples. So, for example, this is an example tumors. We first label with our piezoelectric materials, and this is the labeling, and then expose with ultrasound. We routinely do in the laboratories. This is a tumor spheroids. We make it and then label with our this ultrasound responsive piezoelectric materials, and then expose with ultrasound, and this then check the therapeutic performance, whether cells are dead or not, by conventional biochemical approaches. And this is a results ultrasound-based therapies, tumor therapies. Histotrypsy. This is bubble-mediated destruction of cells or tissues. Bubbles, how you generate? Let's say you make a liquid droplets. This is a liquid droplet nano size, and these droplets are made of some molecules that have low boiling point. In presence of ultrasound, they can be converted into gas. So micro bubbles are formed in presence of ultrasound. So once micro bubbles are formed inside cells, they kill cells by mechanical cell destructions. This is called histotrypsin. So ideally, you need to develop a nano droplets where liquids are present that can be vaporized easily in presence of ultrasound, and they are terminated with some biomolecules so that they can be used for labeling tumors or tumor cells. This is another example where we can design nanoparticles for crossing blood-brain barriers. And this is a kind of molecule that are genus particles or genus or nanomotors or ultrasound responsive particles, gas cores. Gas core, I showed the examples last one. Here it's a nanomotors with a genus structure. Let's say micro nanoparticles, one side is platinum or gold particles. They can quickly degrade hydrogen peroxide. So this oxygen will be generated, and this oxygen can this gas can push your particles in a directional motions. These directional motions leads to nanomotor structures. These nanomotors can be used for crossing these blood-brain barriers. You can also use the ultrasound-based this gas cores. We label cells, and then the cells with these microbubble droplets, and then use sounds to generate microbubbles, and then use this for delivering drugs. Nanotechnology is another powerful approach as antibacterial materials and surface. Nanotechnology-based approach can be used to generate antibacterial, antimicrobial materials and surface. Why do we need antimicrobial, antibacterial surface, antifungal surface? Because the antibiotics that we conventionally use nowadays, they they are resistant. They're becoming more and more resistant. And there are predictions that in next 10, 20 years, no conventional antibiotic will work. So that's why scientists are working on generating new generation antibiotics, small molecule based, primarily organic chemists are working on those. And nanotechnology is another powerful approach. And these are the conventional ways how antibiotic resistance are developed, and nanotechnology approach how we can overcome some of them. One common approach is nano carriers can be used. Conventional antibiotics can be delivered using nano carriers so that they can bypass these antibiotic resistance mechanisms. In addition to that, there are many nanoparticles that generate reactive oxygen species in presence of sound or light, or sometimes in presence of biochemicals. Indigenous hydrogen peroxide present in our body that can also be used for generating reactive oxygens. So nanoparticles themselves can be antimicrobials, antibacterials, and nano surface can be designed in a way how nature did. This is cicada wings. They have nano pillars decorated with wire-like structures. They are kind of 10, 20 nanometer diameters, 100 to 200 nanometer height, and spacings are also similar. So once bacteria are attached on the surface, they're designed in a way that bacteria attach. And once they attach, the bacteria have lots of pores, membranes. Because of these pillar structures, bacteria are attached and there are lots of pores formations. These pores leads to bacterial deaths. So this is called mechanotherapy. So we can artificially design this kind of surface for bacteria, antibacterial surface. Another approach is anti-fouling surface. So these are lotus surface where actually micro nano structural designs so that water don't absorb at the surface, don't attach at the surface. Anti-fouling surface, and water don't attach means bacteria that are present in the colloidal form in the waters, they won't be attached. Anti-fouling surface. So they can be designed in an indigenous way. So I have quickly discussed about the role of nanoscience in biology, how we can design some of the nanoparticles, how we design nanoparticles and nanodrugs for controlling some of the cellular processes, how they can enter into cells by bypassing the endocytosis mechanisms, or sometimes they can enter into cells by endocytosis, but we can use them for different applications after entry. And we also design colloidal nanodrugs for subcellular targeting, followed by therapy, for example, nucleus, mitochondria targeting. We design a lot of sound responsive new generation nanomaterials and nanodrugs for therapy. And sound-based therapy is much more advantageous compared to light-based therapy because sound can penetrate more deep into bodies. And nanodrug also commonly we are designing using principles of nanoscience for crossing blood-brain barriers, and they can also be designed for uh, antimicrobial, antibacterial surfaces. With this, I finish my talks, and let me see if you have questions.
Thank you very much, sir, for your insightful lecture. Already we have gathered a few questions in our uh, message box. So first I need to read one question, and I would like to request to the other participants to raise their hands, those who want to question directly to sir. But before moving to that, I would like to read the messages, the questions rather, gathered already in the message box. The first question has been asked by OP Singh. He asked that, is it possible to conjugate the quantum dots with drugs? If yes, then how can I know how much percentage of drugs have conjugated with drugs in a situation?
Okay, so conjugation is possible. Conjugation is possible, but we usually try to avoid these conjugations because conjugations can disturb some drugs activity because drugs ideally binds with some of the targets, and once it's covalently conjugated with you with quantum dots or whatever nanoparticles, quantum dots are much larger in size. They actually make a steric effect during the drugs binding process. So ideally, what we do is loading of drugs into a nano carriers. It may be nanoparticles, quantum dots, and this loading sometimes, if it is a polymer micelles, nano-sized polymer micelle, we can load them inside, or sometimes they can be absorbed at the surface by electrostatic interactions. They can be loaded in the micelle surfactant interface. This is one options. And other question I forgot. How will
Sir,
how
huh,
will we determine
percentage of
how much they
drugs.
are addressed? So quantum dots, they can be, if they are not very large, we can use centrifuge, high speed centrifuge. After binding quantum dots with the drugs, we can centrifuge to separate quantum dots bound with your drugs and remove the supernatant. That's one option. Other options is we use chromatography. That's called not conventional chromatography like familiar or column chromatography. It's a GPC, gel permeation chromatography, where we can separate different size materials, and using GPC we can actually separate nanoparticles that bound with drugs from the free drugs. Even we can separate nanoparticles drugs versus free nanoparticles using these approaches.
Okay, sir. The second question came from Dr. Orudima Dotto. She is asking that can we directly target the nucleus? If so, what type of NP should be used and what would be the method of NP synthesis?
Okay, we designed some of them. That's how we know it. So conventional endocytosis, cellular endocytosis is a difficult approach because endocytosis require large size, typically between 20 nanometers to 100 nanometer size. So during this endocytosis process, nanoparticles are engulfed by different proteins present in the cells, and then they enter into cells. And drugs, if it is there inside nanoparticles, they will be also trapped in endosomal lysosomes. So you need endosomal escape route. So your carriers have to be designed in a way they have some molecules like amines kind of things, proton sponge effect that can be used for endosomal escape, and drug will be free. But other approach that we use, drug is free means they can, depending on drug's nature, they can bind, intercalate into DNA double helix. That's how they target nucleus. But the other approach, if you don't like endocytic approach, then approach is that non-endocytosis. Non-endocytosis means size should be small. There are theoretical approaches. If size is less than 10 nanometers, that's ideal. Cell cannot use endocytic approach. The endocytosis requires membrane bending, some energy processes. That's not possible. So small size, less than 10, and then you have to have some chemistry, chemical functional groups at the surface so that they can bound with cell membranes, phosphates, via phospho, what is it called? Phosphate, what is it called? Salt bridge kind of interactions using guanidinium groups. So your nanoparticle have guanidinium groups at the surface. They can make salt bridge with your membranes, phosphate. That's how they make a membrane penetration. They enter into cell via non-endocytosis, and this approach will be very rapid. Within 5 minutes, lot of drugs will be entered along with your carriers, and if drugs are electrostatically bound or loaded into your carrier, they will be free in the cytosols. And once the nature of drugs, depending on nature of drug, they can intercalate into uh, DNA double strands in nucleus. That's the one route.
Excuse
Okay,
me, sir.
sir. Uh,
Madam, can I just add something to my question?
uh, okay, just do right quickly. Do it very
Huh.
quickly.
Just in a second. So, as per the chemical composition of the drug you're asking, the question that I asked about this direct nuclear delivery, if these are phytochemicals like phytoestrogens, if I want to use them to control the action on the hormone mediated control of hormones in gene synthesis, if I want to control it, is it possible to deliver certain phytoestrogens by making certain nanoparticles, hollow nanoparticles, maybe the gold encapsulated nanoparticle formation that you earlier you presented in your talk. In that, if I put my phytoestrogen, can they be delivered directly to the nucleus and control the gene functions by intercalating?
Directly means one approach is that you don't use any carriers, nano carriers, directly drugs. Directly drugs usually not possible. Most of the drugs are organic molecules. They are not soluble in water. They precipitate in water. So that's not possible to deliver into cells. And gold, silver nanoparticle, inorganic nanoparticle based delivery is not possible because usually not possible because they are inorganic hard particles. You cannot load particles too much. Maybe you can absorb at the nanoparticle surface. that's not much drug you cannot load. The ideal approach is use polymer based or basic, what is that called, liposome based carriers where
Liposome-based
you can
delivery.
load a lot of drugs. And these are organic like structures, cores where you can load a lot of molecules, drugs. And depending on your phytochemicals, whatever, if they are very small in size, that's easy. If they are macromolecules, again, issue will be there because delivering small molecules is easier than delivering protein like macromolecules because you have to load a big molecule into a nanoparticles. That's also
That
issue.
is not possible.
And case to case basis, you have to design different carriers.
And we have to sort it out by the various methods
Yes,
that which is
not only
better
sort
for.
out depending on your drugs and depending on the where you want to deliver. That also the factor. So basically, it's not possible by biologists itself themselves. You need a collaborator who works on these kind of areas.
Thank you, sir. Thank you.
Okay, so the third question that we have received that coming from Dr. Kazi Manjur Ali, he is asking that are nanoparticles likely to accumulate in the liver or spleen? If so, how can this be avoided?
It's not possible to avoid it. It's highly likely. Nanoparticles, liver, lungs, kidney, heart. These are common places. So that's how inorganic nanoparticles, which are not biodegradable, they usually we don't use for human applications. Only one nanoparticle that's used is iron oxide that I know. Again, it's not biodegradable, and there are a lot of studies that shows they can be toxic many situations. They can generate reactive oxygen species under Fenton reactions and other things. So inorganic nanoparticles, they are not. And for human application, most of the cases is what is the lipid-based nanoparticles or polymer-based, biodegradable polymer-based carriers, nano carriers are used. Whatever commercial approaches, all are polymer or lipid-based particles. No inorganic nanoparticles.
Sir, the next question that we received from Namrata Roy, asking when crossing BBB, do NPs diffuse into endothelial cells?
It's not simple diffusion. Diffusion will not work because entry into any nanoparticles you need either endocytosis or there are certain for some ions or molecules. For example, glucose there are transporters. There are very specific for specific molecules or ions there are these kind of diffusions are possible. But any new drugs or there is no such kind of processes available. You need to use some either use the existing process that are available in cells, or for example, nanocarriers. What we use transferrin receptors. That's one approach, or neurotransmitters that how they enter into cells. Those receptors we use. For example, dopamine receptors. These are most common. Dopamine receptors and what is that called? Another protein receptors. Those are most common. Sometimes glucose transporters.
Okay, sir. The next question asked by Arijit Das. His question is it possible to detect onset of degenerative conditions in brain by using nanoparticles? Also, not just this, but any genetic abnormality can be detected early by using nanoparticles.
Genetic abnormality means single mutation can be detected. There are a lot of methods. Last 10, 20 years, people developed this using gold nanoparticle surface and Raman spectroscopy. There are methods, even quantum dot based methods, because they are very sensitive. But inside brains, nothing is possible. Inside brain, iron oxide, there are examples, extreme cases, patients which are in the late stages. There are examples where people do this kind of experiments, but in actual patients there is no such experiments I have ever seen. It's not possible so far. People use fMRI, functional MRI, for trying to understand different conditions, disease conditions of brains. But these are mostly in research stages. No medical application so far.
Sir, next question asked by Navonita Ghosh. Her question is, sir, are there any nanomedicine to treat Parkinson's disease?
No, no medicines available. Actually, conventional, some other drugs that are used in cancer or some other pain killers kind of drugs are used as kind of minimizing side effects. No drugs are available for any neurodegenerative diseases.
Okay, sir. The next question asked by Prajit Nambiar. His question is, does nanoparticles synthesized chemically and the ones created using plant extracts differ a lot in their functionality?
So plant problem is in plant extract there are many chemicals or biochemicals. If we do not know which one is which one is working, at least primary components, it's almost impossible to predict and reproduce the method. That's how chemists usually don't rely on plant extract kind of materials. Even for a chemist, even they try to use these, they try to understand what is there, try to separate, isolate, and see the main components, and then try to buy those main components from Sigma Aldrich or standard companies and try to reproduce and see whether this is possible or not. So plant extract is more kind of Ayurvedic process. These are not basic scientific approaches. Even in scientific some Ayurvedic processes, people try to extract what science is there, whether we can do some more reproducible science using those approaches. So it's not, in general, it's not that easy.
Sir, next question came from Debolina Sarkar, Dr. Debolina Sarkar. She is asking, what is the component in green tea that can be converted to nanoparticle or any particular compound from green tea along with metal ions are used for synthesis of nanoparticles?
There are many, and this is a polyphenol. Green tea actually, the nanoparticle that I have shown, it's not actually green tea extract. It's a chemical compound, it's a polyphenol that we know that main component present in green tea acts as an what is that called, antioxidant. So we buy this pure chemical from Sigma Aldrich and then use them to make nanoparticles with our polymer systems. So that's how we make a green tea nanoparticles. We know what is there. It's a well-defined chemicals. It's not just green tea extract. We can make nanoparticles from green tea extract, but we cannot tell what is their composition. And it's not possible to make reproducibly. Every batches will be different. That's how we don't use it.
Thank you.
Okay,
Thank you, sir.
sir. The next question came from Rajarshi Ghosh, Dr. Rajarshi Ghosh. His question is, how might AI or computational modeling improve nanoparticle design?
I don't work much on it, but there are a lot of theoretical work on computations on these nanoparticles interaction with cells, biological interface, and maybe there are people have just started working on AIs, how to design them for better membrane blood-brain barrier crossings, what kind of nanoparticles designs and processes required. People have just started working on it. I don't know, near future can only tell. I don't work much on it, and I cannot tell much.
Okay, another question came from Annesha Chaudhary. She's asking, can drug resistance occur against nanoparticle mediated drugs?
Yes, this is a new subject area. Yes, there are few reports, one or two reports. Of course, it's possible. Again, we have to design a different way how to overcome those things. This is a new subject area. That's how that only recently some reports are coming, and near future you will see a lot of reports will come very soon. And at this moment, there is no hardly any nano drug that can use for antibacterial or antibiotic, anti alternative of antibiotics. So what they will come, this issue will again.
Thank you, sir. And now I'd like to Obhik Chaudhary, who raised his hand. You can ask questions, sir, directly by unmuting your microphone.
Yes, sir. Thank you, ma'am. So my question to sir is that you mentioned about diatoms. So can diatoms be used as a template to synthesize nanoparticles? And if so, as their biological entity, living element, so what kind of, say, capping agents or something like that we can use to inhibit the or rather regulate the nucleation process?
So
If
diatoms,
that
people, yeah, diatoms people use it. That not only that we synthetically make this is basically porous silica structures diatoms. So in laboratory we make porous silica. This is a old subject. There are a lot of words. Porous silica, either microparticles, macroscale materials, or sometimes colloidal porous silica particles. This field is very mature. You can also buy some of these porous silica, colloidal silica particles. You can load drugs, functionalize with something on the surface for targeting and delivery applications. But as such, these diatoms, that porous material, they are large, micron size. So you have to design on your own to make smalls so that they can be more useful. The silica is also biocompatible but not biodegradable. So if again they too much enter into body, they are not biodegradable. They will deposit in the body. They may not be harmful to your body, but long term they may be maybe toxic, maybe a issue for the bodies because they will deposit in the lungs.
Thank you, sir. Thank you.
Okay. After just taking the question from Dr. Kuntol Kanti Goswami, I will ask Namrata to ask your question. Dr. Kuntol Kanti Goswami's question is that for therapeutic nanoparticle design, how can we decide whether to target direct entry, lipid wrapped pathways, or clathrin mediated endocytosis?
Okay, there are actually there are many ways, but we as a chemist do their biology. There are different ways, but we do there are endocytosis inhibitors. There are many chemicals. They are known that certain chemicals they can inhibit these processes. Endocytosis like clathrin processes or caveolin processes. Small chemicals, biochemicals are present. We sometimes check them in presence of those inhibitors. If those inhibitors block that endocytosis process, uptake process, from that we indirectly tell that this endocytosis process is occurring in this uptake. So that's how we usually take. But in biology, there are different approaches. They express certain kind of receptors for specific endocytic processes. So they generate certain kind of cells that can have specific uptake process and use those cells for doing the cell uptake, and by doing that they confirm what kind of uptake processes your nanoparticle is doing.
Okay, sir. Thank you.
So now I'm asking Namrata to ask that question you wanted to ask, sir.
Thank you, ma'am. Sir, I'm Dr. Namrata Ray, and I'm an assistant professor of chemistry at Shantipur College. I'm very happy to listen to you because in person also I've listened to you, sir. I've done my PhD from ISCS, from the lab of Professor Amitabh Sarkar, and we worked with nanoparticles. So, sir, the query that I have is, we were able to design antibody conjugated nanoparticles using Fischer carbon complex, and it was a very mild and in situ generation of nanoparticle, and that we used to bind to specific antigens in blood samples and developed a visual detection method using the rose gold color of gold nanoparticles. But sir, this we also tried to use this for detecting certain cancer tissues which overexpress folate receptors. So we generated folic acid bound nanoparticles, and then we tried to target cells like HeLa cells and a few others. But sir, there was first of all a lot of non-specific binding, and there was also a lot of leaching upon washing the first or second cycle. So sir, was it because probably because the cellular uptake was bad because of size, or
Yeah,
what
two
should we focus
issues,
on?
probably two issues. You said the leaching. Leaching is probably linked to probably they are adsorbed at the surface. Maybe they didn't enter into the cells. That's how your washing process remove them. And second is non-specific binding. For that, actually, what we do, we use nanoparticles covered with polyethylene glycol or extra. They are non. They minimize these non-specific interactions. And for example, in surface chemistry, there are people sometimes use BSA, bovine serum albumin
Albumin.
coating kind of thing. So those coatings are important. In addition to that, you have to have your foliate or other molecules at the
Right.
surface. So your ideally nanoparticles should primarily covered with your PEG or dextran
Yes,
and
sir.
then few folate. So that only interaction because of your folate receptors, nothing else. So your design have to be like that.
Right. So the Fischer carbon complex, which we coupled with the folate, was actually pegylated. So that took care of the reduction. Okay, the Fischer carbon did the reduction to generate the nanoparticle, and then that it was stabilized by the uh, incorporated pegylated chains in the Fischer carbon complex itself. So that part we had done, but still, I think there was a problem in the cellular uptake. Obviously, either you mentioned that endocytosis requires a size greater than 220 nanometer.
So
Maybe it
what
was
was your size?
so the size was, I think, yeah, it was 20 to 25 nanometers, sir.
So entry might be
Might be
endocytosis and there may be clathrin or something, they may be trapped.
right.
That is all possible. And also you have to check whether they are really inside cells or outside or in the cell
Right.
membranes.
Okay. Thank you, sir. Thank you.
Okay, we have reached at the end of our session. So I can ask anyone can want to ask any quick question. So for that quick question, I can allow. Do anyone want to ask? Yes. There is one question we have received. Uh, Tondrima Shell asking, sir, is there any possibility to accumulate the nanoparticles within the body after administration as drugs?
Yeah, we have discussed there is high chance if your inorganic nanoparticles or large size particles, they will accumulate primarily in the lungs, heart, kidney, liver. And if they are biodegradable, with time they might some of them or most of them will be leaching. If they are less than 6 nanometer, nothing will absorb at them. All will be excreted, get out of the body.
Okay, is there any question anyone want to know? We have 1 minute in our hand before ending this session. No, no one wants to ask any question. Thank you, sir, for your
Thank you very much.
insightful lecture and the questions people have asked and your patients replied to them. And it's really wonderful for us
Thank
to
you
be
very much.
to with
Thank
us.
you very
Thank
much.
you. Thank you. Dear participants, we are taking just two to 3 minutes break because our next professor is coming to join with us. So please take a very quick break, and we'll come back soon. Thank you very much for your patience.
Welcome, sir. Sir has already joined with us, so we are starting this session. Professor, before I hand over the online platform to sir, would just like to say a few words on sir, and then I will be handing over it to him, the session to him.
Madam,
Yes,
you
sir.
know, I am just, I do not like just a lot formal introduction and all these things. Okay, it's not necessary. People who are working in my field, they know me, and I don't need any further introduction. Brief introduction, I'll make myself only.
Okay, sir. So as you wish,
That's
and
why
so
I've given not given much, only 2 3 lines I've given. So because I do not like that. Anyway, so may I'll just do it on my own.
okay, so I would request, sir, and sir, your time is more or less 80 minutes, and after that we'll have an interactive session where the participants may ask you questions. Thank
Yeah,
you, sir.
yeah. How many participants are there?
There are around 53 participants.
Okay, all of them online only.
Yes, yes, sir.
Okay, but 42 I'm sure seeing, yeah, including
May-
you and me.
Maybe the last moment,
Okay,
someone,
fine, no
more
problem.
or less it was 53, but maybe some of them has dropped out in the last moment.
Okay, okay.
Okay, okay, sir.
So, participants, I'll just share my yeah, first screen. I have to share my screen basically. Yeah. Is it my page is open?
Not yet, sir.
No,
No,
not
sir.
yet.
Okay.
No, sir.
Let's check it.
No, sir.
Yeah, please. Yeah. Excuse me. What is that issue? Also, just wait for a minute. I'll just have a this. तो तुम्हारा तो
तुम करवा दीजिए, किसी को बोल के, सौगत को बोल के आप एक करवा दीजिए।
क्या
प्रसाद जी का एक कोर्स
मुझे पता है,
हाँ।
दैट्स व्हाट आई नो, पर प्रसाद जी को मैं बोल के रखा है।
प्रसाद जी सेड टू सौगत, सौगत पहले हाँ हाँ बोला,
अच्छा
एग्जाम नहीं कराया,
उससे मैं बात कर
मुझे कोई रूट
ठीक है ठीक है, आई टॉक टू।
अगर आज भी वो करा देगा शाम तक एक क्वेश्चन पेपर,
ठीक है, बाद में बट थिंग इज दैट द पीपल हु आर टेकिंग रिस्पांसिबिलिटी शुड डू इट,
दे आर नॉट।
एनीवे शोगत को क्यों देते, शोगत को ऐसे
उन्हें देना ही नहीं चाहिए था।
हाँ,
किसी और को बोल देते जो करा सकता।
अरे चलो मैं बात करता हूँ अभी, जस्ट मुझे
आज से कल में अगर करा देंगे तो फिर हम 5 तारीख से पहले
ओके ओके
ठीक है।
Thank you so much.
I'm not understanding how to share, because share that button.
Sir, excuse me, sir. After that video icon, there is an icon with the up arrow. You click on that, sir.
That's what. There only I clicked, but it is showing that Jimbra search result, and because we go select a tab to share, but what is the which is tab? No tab is coming. So any tab I'm pressing, it's not coming.
Share your enter screen.
Screen is there. Are you able to share? See?
No. There will be one option coming that share your entire screen. You can choose that and then.
Where is that? Pata nahi. Share your entire screen is where? Yeah.
Then press share.
Yeah. Is it okay?
Okay, sir. Thank you, sir.
Are you able to see?
Yes, sir.
Okay. So, to introduce myself, I am Professor Sanjay Ghosh, Department of Biosciences and Bioengineering, IIT Roorkee. My background is basically a chemical engineer. I did my bachelor degree from Jadavpur University, and then finally I am now landed. Finally, I did my PhD from Department of Chemical Engineering, IIT Madras, and thereafter I worked for some universities, other universities and all. Then I am in Roorkee for IIT Roorkee for last 20 years. Now I am basically head of the department of this department, and then I am also HAG level professor. There is a director level professor I am now at this current position. Now, one more introduction is basically that I worked in the area of biofuel, bioprocessing engineering. Bioprocess engineering is my field, and I work for biofuel, then enzyme production, and all other things, different algal products production, and those things I'm going for that. So I'm not going details into my research, these things and all. So here I have chosen a topic is that bioprocess control and system stability. And this control is one of the most important phenomena, or important thing. And many bioprocess, generally people are of all life science people and all these things, and actually many of them avoid this kind of courses. Even BTech curriculum also is for many places it is not there. So I know that it will be unknown to me. I think what is the background of the participants? Is that all some engineering people are there in this participants? Can you interact with me? Are you able to hear us?
Yes. Screen is visible, sir.
Anybody having some engineering background and all?
No, sir.
Yeah, that's the yeah, but that's why the bioprocess. What this unfortunate thing, basically, bioprocess when you are going for production, ultimately you have to give a product out, right? If you do all the whatever research and all you do, and that is limited within the laboratory, then it is the end. No point, because for humankind it will not do any benefit. Only you will produce some papers, and those papers will be out of your in 2 years time, right? No paper is valuable after 2 years or 3 years. So only you will produce those kind of garbage papers, and those papers having no value for mankind improvement and all these things. Ultimately, the objective is to give product, and that will develop the economy. People of the country, the people, obviously people will get job. Many other things will be starting from there itself, and the objective is that government of India is also focusing on that. So, but biotechnology education in India is going in opposite direction. That is my view, obviously, because engineering education, if engineers are not there, nobody is able to take the product from the lab to the industry. Who will take the product from the lab today? If you develop a I mean to say, a very good drug, you have developed a cancer drug, but then a cancer drug you see in the computer screen, your cancer will be okay. If you see that thing in the petri dish or something somewhere else, then it will be okay. Your cancer will be, you will be cured. Obviously not. You have to develop the drug. Ultimately, production. Ultimately, we have to have the large scale production. The first successful product biotechnological product is basically insulin. Human insulin produced by Biocon Industries in Bangalore. It's very famous. That's a genetically engineered organism. E. coli they have used to produce insulin. That's the first product. And obviously all engineered. Even I know, even last this thing, that COVID time, whatever vaccines and all, by large scale manufacturing took place. That is why a Jadavpur engineer only did it, chemical engineering. Anyway, so I have this. My topic is here today is bioprocess control and system stability. A process control is very important because once you go for the process, bioprocess development and all this thing, some production and all this thing, some control strategies you should know then how to control the process, and then only you can ensure the quality and many other things. So the first question that comes into mind is why process control? Now the answer is this enhanced process safety. First is that anything, whenever we go for the process, any industry work and all this, first thing is that worker safety is the first point. The people who are working in the floor, their safety is the first thing, and then process control, any control parameters, if it is under control, then only you can provide the safety, otherwise you cannot provide. Satisfying environmental constraints. Why? What is that? Some say some hazardous chemicals are involved. Say for some process, some hazardous chemical is involved. Say some acid is involved. Some So and if they basically litter around. Control is not there. Naturally, it may overflow, tank, something, anything can happen, and then obviously the whole problem will be there. So environmental, obviously, you cannot control. And what is the kind of gases that you are leaving in the air, and then what is the kind of water that you are leaving in the to the water bodies and all this, that has to be controlled. And then that is only through the control process control only you can ensure those things. Meeting ever restrict product quality. Product quality is another important. Say you are producing, say, tomato ketchup itself, say for example tomato ketchup, but you have to deliver all the time same quality product you have to deliver. Now one batch is coming out this quality, good quality, another batch came out with bad quality, because you have no control. You do not know how much and what is the temperature. So tomato ketchup you are making, so you need to have it heat treatment, and then heat treatment you do not have temperature control. So naturally it will be either overboiled, not boiled, or whatever. Something will happen. So quality cannot be ensured unless the control is there. Then more efficient use of raw material and energy. Obviously, the raw materials that is to be given, obviously have to have the because raw material involves the cost. The cost can be reduced only when you have a very limited money. The amount of raw material that is particularly required, that only is delivered. And the energy cost, obviously, no littering, nothing is there, then energy cost obviously can be saved to increase productivity, profitability. So all these things basically ultimately goes to the profitability. Even some accident and all can be avoided if control is there. If there is no control, then accident will happen obviously, and then obviously company has to pay a lot of money in the medical for filling the medical bills and all these things. So naturally, the company cannot go on profit. So ultimately, a person who is investing money, ये बात ना सबको याद रखना है. So look at somebody who is basically investing money for industry development and all these things. They are not doing for him charity purposes and all these things. If they invest 100 rupees, they surely they want 115 rupees back, right? So that has to be ensured. And unless otherwise, whatever technology you develop, whatever the very interesting things and all these things, nothing will come out in the market. Ultimately, will never give any benefit to any mankind ever. So that is what is to be. You have to keep in mind all the time. Whenever process you develop, whatever things you develop, ultimately think that if it is to be commercialized, then profit has to be there. Otherwise, this cannot be sustainable process at all. Nobody will do anything. Nobody will invest money. They may appreciate, they may give you award, they may give in prizes, they may give something else, but thing is that they will not take it into the production line. Now, what are control systems? Now look at control system. Basically, if you just nutshell, if I want to tell you, they have given you some examples. Say cruise control for an automobile. This is very day-to-day examples and all these things that I have given. But say even if you go to take a shower, so then hot water shower you have to take. So naturally, if the control is not there, naturally your skin will be burnt or it will not be boiled properly. Whatever, you will not get right temperature water and all these things. So many things could be there. Here, cruise control is there. The purpose of a cruise control is to maintain the speed of the vehicle at the desired value despite variations in terrain, hills, that is called disturbances, by adjusting the throttle or the fuel flow to the engine. Now look at what I said, that fuel and all these things. See, if the fuel flow control only is the important thing, unless the right amount of fuel is going to the air, you cannot control this thing. But disturbances always will be there. That is not there in your hand. So always manipulated very well, and the disturbances both will be there. So what is there in your hand is to control the manipulated variables, and the disturbance will be there. By that, disturbance can be overcome by the controlling the manipulated variables. That's what I've given, the cruise control and all this example. Now if you look at this is the control loop that I have shown you. Here one controller is there. So controller means basically I'm not talking about the human controller. It is an instrument, machines, naturally some controlling device. So that is controller is there. And then this called, if you look at the final control element, this one, right? And then the process. The actual process is going on. Whatever process you may have, it may have a bioprocess or a non-bioprocess or whatever process you have. This is the process basically, right? Now, say you are running a reactor site so for large-scale production and all this. This is the process basically. And then disturbance will be there. Disturbance, obviously, there are fluctuations of say voltage fluctuation could be there. All right, so many fluctuations can be there. Even you are using steam somehow, say in the process, in the bioprocess, say you are going for sterilization. You know that sterilization. For that sterilization, you need to sterilize use the steam. The steam temperature can be can fluctuate, and that may not be there because it is coming from the steam generator or boiler, and then there some fluctuation of whatever fluctuations and all that results may come to the steam flow rate and steam temperature. It may affect. So naturally, those things, these disturbances will be there. But in spite of those disturbances, you should have the ultimate variable should be controlled only. That's what the objective is. Now look, that this is the process, and then control variable is going out. This is the control variable. So this is the manipulated variable. So variables will manipulate, and then finally it is going to the control variable, and then this is what is the control variable, that is the output that is coming out, and we have this is the measured value. Look at So this is the measured device. What is this? Okay. So this is the measured device. So measuring device. So measuring device means some sensors and all. This says you are measuring temperature. That means temperature sensor will be there. If you measure the pH, then pH measuring sensors will be there. Whatever measuring device will be there, that will giving a input to the controller. Basically, this is called comparator. Comparator is basically giving input to the controller. So controller having basically two inputs. One is coming from this is a desired value. That means set point value. Say you want temperature. Say I'm just giving an example of temperature. So you want 30 degrees centigrade. So you want to run a bioreactor, right? For whatever purpose you are doing, right? So maybe antibiotic production you are doing, whatever production, and then temperature is, the organisms require a particular temperature to grow, right? Because that is the best, you have to provide the optimum temperature, the best temperature or most favorable temperature you have to provide to the organism so that organism performs the best. And that for that that because this way you are handling the organism, so naturally the temperature best. So that temperature you are giving to the controller, right? Controller is having that information, that is called desired value. Say I want 30 degrees centigrade. Now fluctuations will be there in the environmental temperature. Now look at AC may not work. So in the lab, whatever AC is not working. So naturally fluctuation could be there. Or say in time to time, in the winter, ambient temperature is around 25 or 20, something like that, even below that, even in northern regions, and it goes below 10 degrees centigrade also. So though, and in summer season it will go around 45 degrees centigrade. So environmental variation, those are disturbances, will be there. But what you want, the temperature inside the reactor should be 30 degrees centigrade. Whether outside temperature may be 45 degrees centigrade or maybe 10 degrees centigrade, does not matter. But inside temperature should be 30 degrees centigrade. That is the objective. Then only you can give the best quality PA. And then only the organism will work optimally, and it will perform best, and it will obviously give the maximum amount of product. So that's in the economy can be achieved. So if it is performs not optimally, then naturally it may give less product. So the company may not run in the profit. So this is the control advantage. So naturally you are giving a set value to the controller, and then what is the measured value? Measured value from is coming from the process. Say if there is a sensor, and sensor is giving value temperature say 25, but you want 30 degrees centigrade, right? So your set value is 30 degrees centigrade, but temperature from the process is coming because the temperature sensor is there and is telling you that it is 25 degrees centigrade. So then what to do? The controller will take a decision, right? What to do? So that means controller will take a decision of heating. No, because you need to heat, then only temperature can be raised. Or if the temperature is actual temperature is more, say 35 degrees centigrade, then what? Then process temperature is required temperature is 30 degrees centigrade. You are measuring here 35 degrees centigrade. That means it has to be cooled, chilled. So naturally controller will take the decision, will be taken by the controller, and controller will give the appropriate signal. This is a control signal. The appropriate signal to the what? Final control element. This final control element, that signal, according to the signal, mostly valve, electronic valves are there. Those valves will be controlled, and then naturally, or many other transducers are there. So these are basically transducers, final control element, FCU is called it. And accordingly it will deliver the thing. Say here is steam. Say on the reactor there are two flows are there. One is steam is there, another is chilled water is there, right? If it is temperature is more, then what you have to do? Controller will say that it has to be cooled down. Then what? Then steam valve will be closed, and the chilled water valve will be open, and automatically it will pass through the jacket and all this thing, and it will temperature will burn down. Or if it happens otherwise, so temperature is less and actually required more. So naturally, then what will happen? The steam valve will be open, and the steam also steam valve, what amount it will be open, that also will be controlled. Not only this open or close, not only that, there will be ratio or the kind of things, gradation of that opening. So how much is it? 70% opening, 30% opening, and all this thing. That thing will be decided by the controller according to the control signal, and then it will open, and automatically that steam will come to through the jacket, and temperature will be achieved. So these are how this way any control parameter, not only temperature, pH, and any dissolved oxygen. Say for example, that many organisms requires oxygen, particularly right amount of oxygen is required, and naturally that has to be controlled, because industrial organisms mostly aerobic organisms, so it has to be given. So all this is control loop is there for all parameters, all variables, and all these things. So this is the general one. Now look at how to represent a control system. Now it's a block diagram. This is a very simple answer. Is a block diagram. A block diagram is a diagram that indicates the flow of information around the control system and the function of each part of the system. Such a diagram makes it such easier to visualize the relationship among the various signals to draw a block. So now look, I'll show you how the block diagram looks like. So one here. So now control strategies. So first thing is that control objective you have to fix. Right, control strategies. To fix the control strategy, you have to first there are different points you take care. There is a control objective. That means what? So whatever objective you have fixed, say you want to say cruise control. I have given an example. Say temperature control. This is a control objective. Or pH. The organism behaves optimally at a particular pH. If you change the pH, the organism will not work, right? Or it will malfunction, right? Or it may give some other product. One pH it will give this thing, another pH it will be giving something else. Or for enzyme, say you are doing something enzyme. Enzymatic conversions and all you are doing. So naturally, enzyme also requires optimum pH, optimum temperature, and all this thing, and they have to ensure basically to how you will ensure in the when the large scale production is there in the reactors and all. That is it. So control objective first is fixed. Then input variables. What is the input variables? Can be of two types. One is called manipulated and another is called disturbance. I told you that one is manipulated and disturbance I have shown which is not in our control. That is called disturbance. And manipulated which is which we control. So disturbances we cannot avoid, right? There will be disturbances, but knowing that I have to give the input such a way that is a manipulated variable, such a way that the output is not affected in spite of disturbances. Then output variables are of two types. One is a measured and non-measured. So sometimes we measure the output variable. Here in this case, the temperature control, I showed that after the temperature is coming out, then we measure the temperature and it's giving feedback to the controller. So naturally, this is measured. Another is sometimes is non-measured. So output variables. Then constraints can be two type: hard constant and soft constant. What is hard constant? Hard constant, say a valve is there. You are using a valve, control valve. Now, say steam opening valve or steam close valve or chilled water floating. Now, these valves can be maximally how it can operate from 0% to 100%. Right? Either it will be totally closed, 0%. Right? Flow is 0% and 100% means totally open. Right? In between, you can have any control. Right? 0 to 100%, anything is to 35.5% also you can fix. That's okay. But then, if you hard constant is that say maximum flow when in the full opening condition, say maximum flow rate cannot be more than 10 liter per minute. If it is so, but your requirement is hard 12 liter per minute, then it will not happen. Then control will not work because its hard constant is that it can open full valve. Then also it is giving 10 liter per minute. It cannot go more than that because that is the constant of the valve itself. Then you have to change the valve. So that is the hardware change you have to do. And the soft constant is that you can change where you can. So this is the constraints. Then operating mode. Now operating mode could be batch or could be continuous or could be anything. That is your or cascade. Many other operating mode could be there. Then process safety, environmental constraints, product quality, specification, and process economy. So this is to be you have to say all these things you have to do for this kind of things. And control structure. Finally, control structure can be of two type: feedback and feed forward. Obviously, cascade control is also there. Cascade control and then feedback and feed forward together also is there. So control structure also to be decided. So this is whenever you go for control, there are a few points you have to keep in mind, and accordingly you take the decision. Then I said that block diagram is the important thing. How to represent that I'll show you, but this is a case study. It's simply just giving what I want to maintain level in a tank. Say for example, आपको छत का ऊपर एक वाटर टैंक है and उस वाटर टैंक से आपको पानी आ रहा है, right? Water is coming from the tank. Now the thing is that it can be there will overflow or can be no water at all. So all the water is drained out, and naturally when you go for bathing, you are seeing that no water is coming from your tap, right? Or your pump you have put it on and it is overflowing water, and then naturally your electric bill is increasing. Unnecessary, then the water spilling is there. That spilling may be spoiling your roof also. Then water obviously is a scarcity, scarce commodities that is spoiled, it is unused, right? It's going through the, that's obviously here. So all those things to be, you have to control. Say level control is one objective. This is objective. Control objective is to maintain the liquid level, water level or liquid level. Say acid tank is there, some process, in the process some acid is to be used. So naturally acid tank is there. That acid tank cannot be, you cannot afford it to overflow, because of overflow the acid spilling will be there. Lot of hazardous, environmental hazardous problems will be there. And then control, PCB is also there. Pollution Control Board is there. And if they find these kind of things are happening, then they'll ask you to shut down the company. Simple baat hai. PCB is very strict nowadays. So hazardous chemicals and all you have to control. So so these things. So level control is one of the objective here. Now what is the input? Right. The input is the, look at F1, that is the flow rate. Flow rate, that is inflow rate, and the outflow rate. Both are input. Don't think that F2 is the output. F2 is what? F2 is the flow out of the tank, and F1 is the flow rate that is going into the tank. Okay, so these are the two things, but both are input only. Don't think that one is input and another is output. So input is F1 and F2 both. Then output is what? Output is the of this, because of this flow F1 and F2 flow. What will happen? Ultimately, you are maintaining the level. So that means the liquid height. Liquid height is the output. Then constraint is what? Capacity of the tank. This is the constraint. This is a hard constant. Capacity of the tank. So the tank is 100 liter, right? So, or 1000 liter. So that cannot be increased or decreased. Flow rate can be increased, decreased, that you can control. But tank capacity, you cannot change it, right? Then operating mode, constant continuous process. So continuously what is coming in, continuously what is going out. So that is continuous operating mode. Then control structure, feedback or feedback, and these things I will discuss. So this is look a simple thing. A control, if you want to control the level of any liquid or water in a tank itself, you need to have a whole control structure should be there. Otherwise, what? Spilling will be there. That's why I told you that a lot of wastage will be there, right? And then company cannot afford all those wastage and all these things, because company is there to make some profit. Simple baat hai. Admi is a person who has invested money, thousands of crores he's investing. So naturally he wants some return. And if this kind of things are happening every day and all these things, then what will happen? Company will shut down, and a lot of people will be losing their job. Now look at, this is the control. Look, dekho. Now look at, this is the tank, and then control height. This height is measured, right? There is a level transmitter is there. This is LT. LT means level transmitter. So it will measure the level and it will give. This is the input, right? This input is going to what? This is a comparator. Comparator. I showed you the structure. The comparator, that is this comparator will go there. This is the input, right? Input for the comparator or the controller. Because the comparator ultimately going to the controller. So the FC, flow control device. This flow control device means this is going. So level, what is the current level? Is this is the information going through a transmitter, level transmitter. And then what is the set point? You want, say, total height, you know, that may be 5 meter, and you want it to be set at 3.5 meters. So that is the height point, 3.5 meter you have given the input. So this is the input, and this is basically is the output, you can say, height of the ear. This is how height is going to the ear. So naturally controller will take the decision. So this is FC. FC is the controller. So FC will say that set point is your 3.5 feet, but then actual height is what? 3.2 feet or 4 feet, right? So naturally, accordingly, it will take the decision. If it is 4 feet, more than 3.5 feet, say, then what will happen? So it will, this valve will be more. So naturally, either it will control this valve or it will control this one. Now here, the structure loop I have shown. So naturally, this controller is taking a decision. Accordingly, this decision is going to what? This valve. This is called valve. Then F1 is being controlled, but F2 is left uncontrolled. F2 is not being controlled by the controller. Controller is giving the input to the F1. So naturally, if the height is 4 feet, more than the 3.5 feet, then what will happen? The controller will say that yeah, less flow should be there. So this valve will closed more so that less flow will be there and control. And because output is constant flow, this you are not controlling. So naturally, output is constantly flowing out. So naturally, input by controlling the input flow rate, that you can. So these are the two inputs, but one input. This input is basically, I told you, F1 and F2 both the inputs. So this input is being manipulated, but the other input is non-manipulated. ये input को manipulate नहीं कर रहा. I can control the height of this column tank by controlling this also, this valve also, F2 also. But now I am chosen to control through control the F1 only. By doing that, I can control this thing, height. So if it is more, if it is less, height is less, 3 feet, and then what will happen? So control, I will take the decision. So controller will open it more, so that more flow comes. So that way, ultimately, what is the objective is what? To control the height at 3.5 feet in the tank. Okay, and that is done by the controller. There are obviously different type of controllers are there. This is only, I just give you the overview of this subject, but there are a lot of views are there. Now look at this is a control structure. This is a block diagram that I told you. This is a block diagram. The flow controller is there, controlling valve. The controlling valve, look at this is the controlling. Now which controlling valve, which look at this is the controlling. This is the controlling valve. That means it's controlling what? F1 variables. F1 is being F1 manipulated very well. This F1 को control कर रहा है, controlling valve. And then what happened? Now look at disturbance is what? F2. F2 is what? This is the output flow rate. But control structure say F1 and F2 both are input only. That one input is being controlled, another input is uncontrolled. But this is maybe a disturbance. Says disturbance why? Because if the control, this valve is open more or close more, something happens to this, then naturally more flow will be there. More or less flow will be there than desired. So this is a disturbance. Since I'm not controlling, this is a disturbance. But this is a control being controlled. So naturally, if you look at the manipulated variable and disturbance will be there. Disturbance is F2 now in this case, and F1 is manipulated variable. As a result, the output variable is what? This is a height, H, right? And that output variable is measured by what? Level transmitter. That's what I told you. This is the level transmitter, LT. LT is measuring the level transmitter, measuring the level, and it is giving input to what? Comparator. This is the comparator. This comparator basically nothing but plus and minus. The input is always plus and so set value, set point is always plus, and the whatever value comes from the level transmitter and all these things. Then so this one will be negative, plus this will be plus, and this actually this is the whole course. And then obviously the difference, the error value goes to the flow controller. The flow controller takes the decision as per the error value, and then the controller signal is sending to the control valve. The control valve ultimately signal is going to the, this is a, look at this, all electrical signal only. This is from here electrical signal, level transmitter is measuring the levels, and ultimately level is going in what form? Level is going form in either voltage or ampere form, current form. The level is, that's called transmitter. Why? Because level 3.5 feet, but how the machine will understand 3.5 feet? Machine will understand only in terms of voltage. In terms of voltage or current, nothing else. So this is being transferred, corresponding value is being transferred to the current signal, and this is going to the comparator. The abjo set point 3.5 feet. The controller is understanding in terms of current only. 3.5 feet it does not understand. It understands as a current. But आपको human interface के लिए, human interface will not understand what is volt and all these things. You will give the 3.5 feet input. But that the inside, machine के inside जो है, that inside will understand as a value only, the current value. Now that value is going to this, and then ultimately controller valve is going on, right? Controller, and then takes the decision. Now, have you understood this one? So this is negative feedback and the positive feedback. In negative feedback, the error is the difference between the set point and the measured value. That's why I said the set point is positive and the measured value, that is here from the whatever is coming from the level transmitter and all this is a negative. So this is plus and this is this is plus and this is minus. So that's the negative feedback and the set point and the measure value, and this is usually the desired configuration. Okay. Then positive feedback is the positive feedback. The measured value is added to the set point. Right. This is usually an undesirable situation and leads to instability. So generally, nahi hota, but fast negative feedback is already is a general control loop only works in this negative feedback only. So this is plus and this is minus only. So okay. Now look at feed forward. Now feedback, I told you, feedback is what? After this feedback is that after process is done, then the value is measured and then it is giving the feed to the controller. Okay, but feed forward is the what? This is for feed forward. What it does, it sense कर लेते. मानो this is a tank is there and the flow is there. F1 and F2 are both there. Now look at when flow is there. This is happening in the activated sludge plant and all these things. So water, आपका जो municipality water treatment, waste water treatment plant and all, they do these things. Basically, initially the more flow is coming, say from the municipality water and all these things, more flow is coming. Then immediately sensing this flow is coming more. So do something before it goes to the ASP. So then it goes to the this is this one. This is going to the FFC, flow control. So this control device information is going, and then automatically take the decision here, and accordingly decision is executed through the valve. So this valve is being controlled. So F1 is not being controlled. F1 से information जा रहा है. This information is going to the controller, and then accordingly it is controlling this valve, so that that means they are controlling now F2, not F1. Now look at here. This is the block diagram. Disturbance is now F1. So F1 is the disturbance. Look at, because F1 is not controlled here. Previously F1 was being controlled. Now F1 is not controlled. So this is a disturbance. So F2 is controlled. So this is your manipulated variable. Now this is disturbance. This is and the flow measurement is there and is going to the this feed forward controller. This controller में information जा रहा है, and then is going the signal is coming to the control valve, and then it comes the so manipulated variable is what? F2. Look at here, for manipulated variable is F2, not F1, and then is going to the process. Ultimately, control variable is H. This is the output, but output is not measured. Look at here, output is not measured. Previous loop you look at. So output is measured here. Look at output is measured here. Level transmitter is there, but here for output is not measured. Look at here. So this is the ultimate. Now here, this is one more I have shown. This is feedback and feed forward both. The feedback I have shown you, feed forward is I have shown you, and then feedback and feed forward both. So what is happening here also? Out of two inputs, F1 and F2, F1 remain as disturbance and F2 remains as a control variable or manipulated variable. Now look at, but it is measuring, this is a feed forward loop. Feed forward loop, that means it's measuring flow measurement. FM means flow measurement. It is going and giving the input to the flow controller. Now controller को पास और भी two inputs है. One is your set point. Another is your level transducers. So controller has three inputs in the sense that one is set point, another is the output, and another is information from here. And accordingly, it take the decision and ultimately going to the this decision is being executed through this control valve. So control valve is very important in any process. So ultimately, is being controlled. This is all electronic valve basically, not that manual one. So automatically it gets controlled. And you see the same block diagram. This is the actual process I am showing you, and this is the block diagram representation. So this is look at F1 is disturbance and F2 is basically look at this is F2 manipulative variable. And then so look at so controller का पास क्या information है? Set point is there, then measured signal is there, and also input is there from the flow measurement. So 1, 2, 3 inputs are there to the controller, and then accordingly signal is coming, and then valve is being controlled and process is maintained. The process is basically here is the ultimately height control. So this is this. Okay. So block diagram you have understood. Now how to do this execution, how to represent the dynamic characteristics of the control system, because this is not static one, dynamic characters. Okay, now bit of mathematics will be there. Right, this is I told you the issue, the problem, and how you represent the problem. Right now, obviously, transfer function. The main thing is the transfer function, how you generate. This S and T. There are two domains are there. One is Laplace domain, another is the T domain, time domain. You know, Laplace domain is represented by S, and time domain is represented by S. I think Laplace transform many people have done. I do not know if somebody has done mathematics in engineering mathematics have done. Then Laplace transforms you have done. I do not know how many of you have done. Chalo, that is. So transfer function is what? This y by x. What is y? Y is the output, and x is the input. Right. So this is x is the input in deviation form. Again, x is the input in deviation form. That means what? That means the x minus xs. Now that means steady state value. So what is the steady state value? So whatever difference is there, that is the deviation value. That deviation value is given as the input, right? And this is similarly y as also a deviation form. So both are deviation form basically. So y is the output, and then x is the output, and the ratio of the output by input is basically transfer function. This function you have to, if you can find out the function, then control loop is done. It will the machine काम करते equation को समझते. It understands the equation. Your language, English language, it will not understand. Equation has to be transferred in form of signals ultimately. So how mathematics works basically here. You see the now look at I'll go details into that. Look at this is that means y. Now look at this y gs is equal to g is equal to y by x. S in bracket that s I have given, that means you are working in the Laplace domain. Whenever I am putting s in bracket, it's called understand you are working in the Laplace domain. If you put in the t in the time domain, so s and t the conversion is basically a Laplace transformation. That many people those who have learned engineering mathematics, they know. So g is equal to y by x basically. Now that means y is equal to what? G into x. That's what I given in the next one. Y is equal to g into x. Look at this x is the input, g is the transfer function. So input is transferred by the transfer function to output. So y will be equal to g into x. Very important understanding. The output is nothing but how you will get the output when the input is transformed by this transfer function, by the transfer function g. That you have to determine the g basically. Once you know, because you know the input, you know the output. You know the to get the output, you should know the g basically. Then you can get the output. So okay, function now. Now this input also called a forcing function, and output that is called response. And g means transfer function. This is schematic representation, and this is basically expressive. So transfer forcing function by the way of transfer function is going to the response. Now this is some of the Laplace transform. I do not know how much you will understand. Yeah, the unit functions you have, that is unit function, that is 1 by s, unit step function and all this thing 1 by s. Some at, that means your ramp will be there, 1 by s square transform, and then obviously and the vice versa will be there. If that means 1 by s is there, they will look at 2 t likha hai. This is a t domain, and there is a corresponding Laplace domain. T domain means time domain. So u, tu, all this that you look at in bracket t likha hai. And here you see the second column, you see the all are in S. That means if this is the time domain function, then corresponding Laplace domain function is this, right? So and vice versa. That means if the Laplace domain function is this, then corresponding time domain will be like this. So we do it like by taking log and anti-log. You take the log, do it, all the process and all the log mein multiplication will be what? Like addition. Division will be like what? Like subtraction, right? All these things are done. And finally, whatever you get value, log value you got, you do the anti-log and you get the actual value. Similarly, in the time domain, what you do, whatever values are there, you transfer to the Laplace domain and do all these things. Finally, we again convert it to this Laplace domain to the T domain. That's the same thing, log, anti-log kind of things. And these are all other functions. So these are the different type of functions. Cos, this is hyperbolic sine, hyperbolic cos, and then exponential function, and many other things. Pulse, unit pulse. Now say you have injected a very in the reactor. Say you have injected some vitamin solution, right? So what will happen? So that is also will go as an impulse. So when you are, if you look at your body function also, so आपका जो है body, you are gone to the hospital and all this thing. They are giving some injection, right? Doctor is giving an injection. How you can mathematically look at? Mathematically you can look at is impulse, unit impulse. Unit impulse is right, and then it's going to the system. Your system is now flowing dynamic system, right? And that system can and system is giving there's input. So input is basically an injection, and the output will be system obviously is responding to that. That's why doctor is pushing you the injection. So this is the unit impulse. Or you see in the reactor you are putting something at very short interval, very high dose. That is called impulse. So you are giving vitamin solution. All so some reactor is on, and some at certain point of time you decide that you should inject vitamin solution, and then all the things will go on. Whatever you decide, that may be impulse. So you need step input. Step input means what? Mano, you are working at say 30 degrees centigrade temperature. Now you want to shift it to 35 degrees centigrade. So input is your 5 degrees centigrade input increase. So that you, and then you are maintaining the 35. So initially it was 30 degrees centigrade, and then all of a sudden your chilled water. Say chilled water was coming at 10 degrees centigrade, right, in the reactor. But somehow, some reasons and all this, and some people are changed the chilled water system, and is now giving 15 degrees centigrade water is coming. Now it is a step input. That means 10 degrees centigrade to 15 degrees centigrade, step input. And accordingly, the system will also respond to that. A system always will be responding whenever changes are there. If it is a living system, if it is a vibrating system, it will always respond. Then how to write transfer function? Now here पर आप देखो, so I do not know. I never look at a clock for giving lecture. Chalo, by you. As a standard block diagram, now look at this is the GC is the transfer function of the controller. G1 is basically transfer function from the final control element FCE. And then G2 is the transfer function of the process. So whatever process you are doing, you can have a transfer function. Whatever, it is a biological process, it can be chemical process, it can be any process. There will be some transfer function. That process can be represented by a function. So that function will be G2, right? And then you are using a measuring device, measuring sensors and all these things. That also will give a transfer function. That is the H is the transfer function of the measuring device, right? And then all these transfer functions. And look at U is called as disturbance, and R is your input, right? That means set point on all these things, and C is your output. So you can say R and U both are input. R and U both are input, but U is unmanipulated. It is not manipulated, and R input is manipulated. So this is manipulated and manipulated. This is M is a manipulated variable. M is a manipulated variable. So ultimately C is basically the output is combination of this disturbance is also there, and the input is also there. The set point is also there. So all these things are there. So So, and ultimately this is being controlled and other is not being controlled, but ultimately C has to be controlled. Then this is H transfer. So all the transfer functions are given, and then error. What is this A? E is basically, look at here, I have given plus and minus. The plus is the set point, and the minus is what? The measured value. Whatever measured value coming here, that is E. So the E will be equal to the R minus B, basically. E is equal to R minus B. So the difference is going to the GC controller. Controller. So there are proportional controller is there, proportional integral controller is there, proportional derivative controller is there, proportional PID controller is there, proportional integrative and derivative controller is there also there. So many controllers are there. I'm not going to details into that, but they're just showing the control loop. So now look at overall transfer function. There's a closed loop transfer function, right? Once a control system has been described by a block diagram, the next step is to determine the transfer function leading to C to R or C to U. So that means what? This is because C is the final output, and C to R, that means how C will be responding, the output will be responding in corresponding changes in the input, right? And how the C will be responding corresponding changes in the disturbance variable U. So C to U ratio, C to U relationship and C to R relationship to be found. That means मैं change कर दिया, मानो set point मानो all of a sudden मैंने reactor का set point change कर दिया 30 से 35 degree से change कर दिया. Now system obviously will respond, and then how the system will respond, that or say you have taken as the productivity. Productivity, say your antibiotic production you are doing. So what is the productivity? That productivity equation is there, that productivity will be changed. So because reaction जो है, that reaction parameters are changed because Arrhenius constants is there, obviously different. Reaction constants will be different because temperature is changed. The moment temperature is changed, actually all these things parameters are changed, then the reaction rate is also different. Reaction rate different from the productivity also will be different, and that productivity is the output, say for example. So whatever you decide. So that is the So now look at now look at this is one. You have the now we have two things I've taken. One is the no disturbance is there, U0, but only changes is there in the set point. Changes is there in the set point, but no changes in the disturbance. Disturbance कोई कुछ नहीं है. U is equal to 0. But then you see how the control loop looks like. That means relationship you will get between C and R. You will get relationship between C and R. Now what GC is the control of transfer function. G1 is the function final control element transfer function. G2 is the process control transfer function. H is the transfer function of the sensor. So all these things are there, and ultimately C will be affected. Now look at how every function, if you look at, then ultimately this GC, G1, G2 can be clubbed together to say G. G is equal to GC into G1 into G2. All the transfer functions together, and then C to R ratio, and then H is transfer function. Now look at this is forward loop transfer function is G, which is product of G1, G2 and GC, G1 and G2, and the H is only alone. So overall transfer function will be that I will be showing you the calculation. C will be equal to C and R. So this is this is the output and this is the input, and this is the transfer function. G by 1 plus GH is the transfer function. So that means how do you get C? C is equal to R into this transfer function. How you got this? Just simple calculation, very simple calculation. X by E is equal to GC, because look at this is GC. This is for this one, for this alone, the X is the output and E is the error is the input. So X by output by input will be equal to transfer function. So X by E equal to GC, or you can look at X by E is equal to GC. Similarly, M by X will be equal to G1. M is the output and the X is the input. M by X will be the G1, right? And then similarly, C by Y will be G2, and look at C. Here C is the output and Y is the input. So this input again, Y is equal to what? This plus this, M plus U. But U now 0. In this case, U is 0. Y is equal to actually what? Because दोनों plus. Look like this is plus and this is plus. यहाँ पर देखो, this error plus and this is minus. So E is equal to what? R minus B. Look at E is equal to R minus B. But whereas Y is equal to what? M plus U. In this case, U 0, so Y is equal to M only. Now say C is equal to C by Y is equal to G2. So this thing is C by Y is equal to G2. And then B by C is equal to, now look at, this is the B is the output and C is the input. C is the input and so B by C is equal to H. B by C is equal to H. Now if you all these things, if you take together, U 0, then look at Y is equal to M plus U and U 0, so Y is equal to M only. And E is equal to R minus B. Now if you take that, if you multiply these things, C by E, relationship find, now what you have to find out? C by E relationship. C by E, how do you get it? C by E is nothing but X by E into M by X into C by Y. C by E relationship what? X by E into M by X, X cancel, and then C by Y. You understand? So, no. So Y is basically nothing but M basically. Y is nothing but M only because U 0. So this C, Y is M, so M cancel, X cancel, C by E you are getting. So to get that, X by E is GC, X by M by X is G1, and C by Y is G2. So that means C by E is nothing but G. Now C is equal to what? G, and E is what? E is equal to nothing but R minus B. So GR minus, and B is nothing but what? B is again, B is nothing but C into H. Here, but look at B is equal to C into H. So you put it there, and then if you rearrange and then get the ratio, C by R is equal to G by 1 by GH. So this is basically, this is the relationship finally you got. Look at this, is C by R is equal to this transfer function, or C is equal to R into this transfer function. C is the output, R is the input. So that means how the output will be responding for a change in the input. That means say 35, say 30 set point you have all of a sudden changed. Say for example, water जो हो रहा है, that water bath का जो है 30 से 35 भी कर दिए. How the output will be changing? C by R ratio is important. That is this is your transfer function. G by 1 plus G. So that G is basically what is the product of all the transfer functions. That is controller transfer function. G1 is what? That control element, final control element transfer function, and G2 is from the process control transfer function. So all this control transfer function comes together in G, and H is a sensor transfer function. Now here the reverse I have done. I have now put R0. I'm not changing any set point. Right? Set point is not changed, so the R is 0, but disturbance is there. So R is 0, disturbance is there. Now what is the relationship you will get? You will get between C and U. Right? Because previously I got C in R because R was there, but now R is not there. R is 0. So if there is a disturbance change, disturbance which is not controlled which is not in our control, basically something happens. Something happens. So कहाँ पर भी कुछ भी हो सकता है. That may be U, right? And that is not in our control. But then how the control function, how the loop will respond to then look at C is equal to what? Now look at G2 into Y, and then Y is what? Y is nothing but M plus U. So C is equal to G2 into Y, and Y is nothing but U plus M, right? Y is equal to U and M. Look at U plus M. Okay. Then M is what? M is nothing but GC, G1 and A. Right? Because M is nothing but M by X is equal to G1, and then double multiply. M by E will be equal to what? GC into G1. Okay. So this is done. And E is equal to what? E is now look at R minus B we write. But R is not there because R is now 0. So only minus B is there. E is nothing but minus B. So now if you all this thing manipulate and B is equal to nothing but HC. That I have given previously also. So now what? Now U and C relationship will get, that is G2 by 1 plus GH. So this 1 plus GH is basically, this is your characteristics equation. And this is the main thing. 1 plus G2 is basically, look at here, the process only. Process transfer function. So C by E relationship is nothing but G2 by 1 plus GH. Look at when C by R relationship was there, Look at G by 1 plus GH. दोनों में फर्क है देखो. And G is nothing but what? G is nothing but G1, G2. Yeah. G is nothing but G1, GC, G1, G2. All product of all these things. Here, look at, and when C and U relationship is there, C and U relationship is there, then G2 by 1 plus GH. But the denominator is the same for both. Now, if I change both, so a single case I have been in first case there I have put R change, no U change. Second case, no R change but U change, right? Now I'm giving the third case when R and U both changes. That means set point also you are changing and then disturbance also may come, right? So you do not know anything. So आपको जैसे करना है. So now look at then what C will be basically one commutation between C and R relationship is this G by 1 plus GH, and U is coming from disturbance and all this thing. So C is nothing but what R and U ka so R into transfer function. This is R into transfer function that is coming to C, and then U into transfer function of this that is coming to C. So both are added up. Simple algebra is involved. You can look at G is nothing but GC, G1, G2, whereas C and U relationship is G2 by 1 plus GH. Simple algebra is there, only just to do the algebra you can find out. So yeah, algebra basic things I have given. C is equal to G2 into U plus M, and U is an right. M is nothing but this, and then E is nothing but R minus B, and B is nothing but HC, and then all these things put together and C. Then C expression, you want to get the EC expression, then you will get this. So that's the output. So output, how is it? So that means this output will be changes if R there is change in the R, or there is change in the U, or if there is both changes are there, then also it will be effective. So C will be affected by both. Right? That means the output level transducer, that means level height in the tank will be affected by what? If there is set point change, that means say 3.5 feet, say 4 feet up change kar diya, set point change kar diya, that is the R. Or there is some disturbance change. Now the control valve which is not there, that means now say F2 may change ho raha hai. So some kind of changes are there, some kind of fluctuations are there, or somebody inadvertently open kar diya. मानो कोई अननोन वर्कर आया प्लांट में, वो देखा नहीं समझा नहीं, and he just opened the valve. He did not understand. So that is also disturbance. So then how the things will change? So then immediately they will give their response. Same thing actually even economy in the economic analysis also people do. Even the RBI also, Reserve Bank of India, changes the CRR ratio. Then that repo ratio, repo factor, they change. And then they want to see that how the Indian economy changes. So this is the input changes, and they want to see. Then they study. Lot of studies. They after 3 months, 6 months they study. Or they on the repo rate is changes, right? 0.25 is changed, and then they want to see how the Indian economy is reacting to this change. And then they study and find the output में क्या change हो रहा है because of this change in the input. So they study all those things. Same analysis, kind of analysis they do. Okay. So generalized for the single closed loop feedback, I do not know how much time gone. The time game मुश्किल होता है. Okay. Now y by x is equal to pi forward by 1 by pi loop. This is negative feedback mainly works, so that I am only focusing. So y is the final output and the final expo, x is the input. Then pi forward is the product of transfer functions. Pi always stands for product, right? And sigma stands for sum. So product of transfer functions in forward path, and then 1 by pi loop. Loop is basically the total path, product of transfer function in feedback path between the location y and x. So full pi feedback. This is pi feed, and the pi loop is basically total one. Pi forward into pi feedback. Pi loop. This is the pi loop. The pi full. Well, that I have shown you that already. So this is finally just to summarize. Now there shall be cascade control also. Now control का अंदर control होता है, right? Say this inner loop is there, and then say there are two disturbances are there, U1 and U2. There here are two disturbances are there, and one reset point change could be there. And look at GC1 is a controller transfer function of the overall loop. But thing is that inside there is another loop is there, which is basically again transfer function. GC2 is the transfer function of the controller in the inside here. And that is big, and then the process one is G1 and H2 is the sensor, and H1 is the overall one. So now look at this one, this plus here for manipulated one. This will be equal to GC1 into this whole thing, and into this whole thing is what that you can find out, and that will be multiplied by GC1. They will be giving to this output, and then ultimately this. So this one complicated example. There can be more complications could be there. So H1 is the transfer function from the sensor of the overall one. This one this process whatever, and then H2 will be again that one. And GC1 and GC2 are the transfer function of the sensors. Sorry, not sensor equivalent controllers. And H1 and H2 are the transfer function of the sensors. And G3 is the one process, the overall process, and the inside process G1 transfer function. So if you look at all these things, and E1 and E2 are the disturbances. Now this is this. Look at how it is coming. Look at now GC. This is your GC1 outside, and the inside whatever this one is there. This inside this loop, this transformation overall transformation will be like this. This one. This is GA. They represent GA. GA represented by GA. And this is nothing but GC2 into G1 by this thing. So this is whole thing. GA is representing the transformation of the overall loop, right? And then GB is basically the transformation of this one, right? And then H1 is. Look at how the C will be affected. C will be affected by if there is change in R, or there is change in U1, or if there is change in U2. So disturbances. There are two disturbances are there. So there any disturbance change is there, it will be affecting the C final output, or if there is a change in the set point, also there will be. So how it will be looked at? This is the first one, GC1 GA into GB divided by this whole thing, and here the same thing. The bottom one is the same. Bottom one is the same for all. Upper mein difference is there. Look at first for one here GC1 into GA into GB. Here for only GB. And here for only G3, because GB is what? Look at GB is nothing but G2 into G3 of the U1, and when U2 is there, only G3. Because C and U, look at C and U2, what is the relationship? There is G3 only is there, but C and U1 is what? C and U1 का अंदर क्या है? G2 and G3 both are there. So look at this GB is nothing but this ऊपर में GB is nothing but product of G2 and G3, and this U2 is only G3. Bottom to same. So all these things are plus together. So C. So U1 is also not 0, U2 is also not 0. So if U1 is 0, then this is not there. U2 is not 0, it is also not there. Only R will be there. R and C relationship will be there. So whatever, maybe anything, if R is 0, then this is not there. That means R is 0 means what? Physically means you have not changed the set point. Now stability. Stability is the control loop. मैंने बायोरिएक्टर पढ़ाता हूँ, so I teach the bioreactor. So there continuous this context is very far from now here. So I show the dilution rate. I know there is a residence time, and the universal residence time is dilution rate is called. So I show the students that there is one point where the reactor will run most optimally, but that point is most vulnerable. Reactor जहां पर सबसे बेस्ट फंक्शन करेंगे वहां पर ही रिएक्टर सबसे अनस्टेबल रहेगा रिएक्टर. If there is a fluctuation there, and then the reactor will be over. You will not get anything from the reactor. Right, afterwards you will see the 5 days you have done for antibiotic, you see that no antibiotic has come. Only thing pani, what is only coming out, nothing else. Why? Because there was a control system failed. Because there is a point. I cannot show you here. There is a point where if you go beyond that point, it will go automatically go to the it will be drained. That means washout condition में पहुंच जाएंगे. I think जो biotechnology student people have bioprocess engineering, if they have a course and all this thing, washout I think they have taught. So it will go to the washout position because the point which system is most unstable. If at all for any fluctuation, if there is it goes to right hand side, then it will never come back. The reactor never will come to the same position again. You cannot stop its draining. You cannot stop its washing out. It will automatically wash out. You cannot stop. So that's why the control is so important. You have run a reactor for 5 days, 6 days, and for continuous reactor and all this. Ultimately, if you see that nothing is coming out, because of you do not know the control loop, then what will happen? Computer next day निकाल देंगे उस क्या ये को, right? Fire. Now look at here, stability is most definition of stability. Okay, so I have not told you one thing, that is about the controllers. So controller transformations, lot of things are there. उसमें I just मोटा मैं बोल देता हूँ. जो there transformation GC is there, that controller function transformation of the controller. There are 3 types of controllers are there mostly, largely. One is called proportional controller, another is proportional PI controller, that is proportional integral controller. Another is proportional PD controller, that means proportional derivative controller. Another is called PID controller, that means proportional integral and derivative controller. So all these 3 controllers are there, but their transformation, how it comes and all that, I am not have not put here. Now I'm just going straight away from this point to the stability point. How the control loop will be stable, that means where the system will be under your control only. Now look at here, GC1, GC value I have told controller transformation is I have given as 1. Okay, and then I have given this is the G1 is the transformation of the control element, final control element FC. So their transformation is given 1 by S plus 1. This is in a Laplace domain, remember, right? So this is a Laplace domain I have given. This I cannot go details into it because the mathematical structure is there before that. So I hope that something you will understand. So and G2 is the transfer function of the process, that is 1 by S plus 1 in Laplace domain. So all are in Laplace domain. And H also I put for simplicity's sake, H is equal to also 1. Because this controller could be 1 lagaya, H is equal to also 1 lagaya. Because this is your measuring element transfer function. That I am just overlooking. What is important is that the process transfer function, the most important one. Another is the final control element, जहाँ से control होता है. So that is. So anyway, so and this U is the disturbance, R is the input change. Now this is this. The characteristic equation is this. Now look at C is equal to what I told you, that C is equal to मैंने ये जो देखा है, C is equal to previously भी देखा है. Look at C is equal to what? R into. So look at C is equal to what? R into G by G plus 1 plus GH plus U into G2 by 1 plus GH. Is it okay? C is equal to what? C is equal to this plus this, right? This is what? This loop is what? R into G1, and this loop is what? Here पर this loop is what? U into this one. वही मैं लिखा यहाँ पर. Here C is equal to this into R1. This is GC G1 G2 by this whole thing GH. This is a G by GH, right? And here पर you look at the G2 by GH. So look at this is called the bottom equation is called characteristic equation. That I'll take it later on, right? And G is nothing but the overall transfer function of the GC, right? Now R and U both are there. That means disturbance is also there, input change is also there. Now just to make simple, GC G1 G2 GH, G2 and H. So product of this I call as G. right? The G is nothing but what is that? Total product of CGC. This is G1, GC into G1 into G2 into H. All these things product I am calling as G. So this GC G1 G2 by 1 plus G into R, and here for G2 by 1 plus G into U. So this is your C. Now look at, now if I put those values, now what is R? R is a step change in the input, right? That means set point change. Set point change mano one step change, say 5 degrees centigrade I did, right? Temperature. So 30 say 35 jana hai. So that means what? 1 by S. Now 1 is what? That's 5 degrees centigrade. Now 5 is considered as a unit. One unit is, one change will be either 5 or 10 or 15 like this. So 1 unit is 1 by S. Okay, so R is equal to 1 by S. Now, okay. And then U also a step change. So now U is considered as 0. Right? No disturbance is there. Only set point changes is there. So no disturbance, that means this part is not there. Only C is equal to this into this. Okay. So 1 by S, and look at, then if I put all the values here, that corresponding those values I have shown you, those things if I put, then this kind of equation will come. I'll come to details. And this is this S minus R1, this R1, R2, R3, Rn are the roots of the equation. So now this is the characteristic equation is basically, the characteristic equation is 1 by 1 plus G equal to 0. This is 1 plus G. So my bottom part is always the characteristic equation 1 plus G equal to 0. 1 plus G equal to 0 is the characteristic equation. That will tell you the character of the equation. Whole thing, the whole process will be how they're stable or not stable. That can be analyzed from this equation only. Now this is the मैंने देखा जो मैंने I have taken the input 1 by S1 and 1 by S1 here पर G1 is equal to 1 by S1, G2 is equal to 1 by S1, and GC is equal to 1 and H is equal to 1. This I put it here, right? And then I am getting this one, right? And here two step change is there. 2 by S देखा है. 2 by S, that means 2 step change, 2 unit step, or you can make it 1 step. Unit step means 1 by S. It does not matter. It will 1 unit step 30 से 35 किया है. That means 1 unit step हो गया. Unit, how you define that is depending on you. Say you say the 2 degrees centigrade is like a 1 unit. That you consider. Okay. Then 2 unit step change means 4 degrees centigrade change is there. Okay. So that is up to you how you define. So this is this, and then it is the equation coming, and you will see that all this thing, that if you just take the algebra of it, it has no big mathematics is there. So all these things, if you take the S1 plus S1, S1 into S1 will be the LCM, and all these things if you do, then something will come. यहाँ पर जब मैंने देखा ना FS, this is G1, G2, G3, you can understand. This equation, ये जो equation जो है ना, this equation, this equation, when I put those values, then things will come, and that they will come with this root. This kind of equation will come, S1 minus S minus R1, S minus R2, and S minus R, and then upon me FS. This FS is what? That I have shown you in the next slide. Here, this Sn is this S1 plus S2 and all this. Because यहाँ पर जो equation है ना, this equation simplify करो. Just algebraic simplification. That you do, you will get this. That is the red one is Fs. Okay. Now, if you have this equation, now this one is S plus 1, S1 will cancel. तो ऊपर में क्या रहेगा? देखो, this S plus 1 and this S plus cancel. S plus 1, S1 cancel. Then ऊपर में रहेगा 1, and if you multiply this one, S square plus 2S plus 1 plus यहाँ पर 1, so 2. So S square plus 2S plus 2. This is a simple equation. ये जो पूरा equation को लगा के तो ये, and then this will be 2 by S. Or if you say 1 unit step change is 1 by S. So this one equation, now if you, this is C, because S domain, still it is a S domain. That means Laplace domain is there. And then आपको partial fraction method, already you have learnt in 11-12 mathematics itself, partial fraction method you have done. Even calculus also, integral calculus you have done partial fraction methods and all. So that is used to separate. So A by S and B into, so B and C to be calculated basically. So A, B, C are the constants. This A, B, C are the real variables. So that has to be calculated. So now ultimately, if you look at, if you do this here, ABC, if you calculate, then you will get like this. So 1 by S minus S plus 1 by this one, and this is this. Now look at, now ultimately C is equal to these equations you got, right? Now if you, now this is S domain, and now if you convert it to a T domain, then CT will be equal to what? This 1 by S is equal to 1, right? And then this one, that I've shown you, that Laplace transform and all these things. From there, if you go back from the now Laplace domain to the T domain, if you go back, you will get this. So this equation in the Laplace domain and this equation corresponding time domain, you get this. Okay.
Sir, can I please interrupt?
Yes.
Sorry, I'm very sorry, sir, but I think we have just 5 more minutes. So can you
आप
please?
लोग टाइम में पढ़ा रहते हैं, इसीलिए मुश्किल है. मैंने जब लेक्चर लेते मैंने when I give lecture in my IIT, I always take a last slot. Why I put the last slot? Because I know that if both sides are bounded, this side is also, then this side is also, cannot be emotionally you are teaching and then you cannot complain. यहाँ पर हो सकता है. I can stop. But
No, sir. You can. You have still some more minutes left. But please, can you please means conclude in just 2 to 3 minutes more?
Time to bound हो जाता है, but then thing is that lecture cannot happen like
Yes,
that.
I understand.
But then only.
Thank you, sir.
People will be sacrificing few minutes from their lunch. That is important. If they are wishing to learn, and if they want to get a certificate, then give them a certificate without learning anything. You have
No, sir.
to give a certificate. Mostly people are
You can
interested to get
continue.
certificate only, not to learn.
No, sir.
जैसा
I think we can
हमारा भी
manage
बीटेक में
for a
हमारा
few minutes
बीटेक
from
स्टूडेंट
now.
आता है, they are only interested to get CGPA and nothing else, and finally a pay package, that's all. They are not interested to learn. Most problem is here, and most participants in this kind of courses and all, they are only interested to get certificate, that's all.
No, sir, you can continue.
Anyway, chalo,
We can.
I can tell you, okay, just in a nutshell, I just 2-3 minutes, I'll just finish it. Now, this I have told you the characteristic equation. This characteristic equation का जब solution होंगे, when you give a solution, do the solution, and that solution, if you look at this, is called argon plane. Argon plane is that plane, it's not real plane. Understand? Real plane is that where x-axis and y-axis both are real number. Argon plane is that plane where x-axis is real axis, but y-axis is imaginary axis. Look at here, this is the real axis, this is imaginary axis. Then that plane is imaginary plane. Sorry, called argon plane. In the argon plane, if the solution comes to the left hand side of this, then the system is stable. If any solution comes on the right hand side or on the lies on the, look at this, these are roots. Roots means solutions. Quadratic equation हो सकता है, polynomial equations हो सकता है, there are more S4 polynomial equation will be there. Then if polynomial equation 4-degree polynomial is there, there are four roots you will get. And if all the roots are lying in the left hand side of this here, that is called then the system will be stable. But if it goes to the right hand side or lies on the imaginary axis, solutions lying on the imaginary axis, then also system will be unstable. So that's the mota baat ye hai. And how it looks like, if the solution falls on this axis, imaginary axis, then it will be a unstable region, obviously, and there will be fluctuation like this, but this is bound. Okay. But here this is unbound. Look at all the solutions here, this side, the right hand side will be the fluctuations are there, and then it is unbound. Right here also fluctuations are there. So on the axis it will be fluctuation but bound, but no, it will not reach to any definite value. But whereas if you look at the values in the left hand side, you see there is fluctuation but stable, because ultimately it's reducing. The frequency is the magnitude is reducing and ultimately going to the stable value. Look at this one. Right here also it is going to the stable value. So all these things, all solutions are there, it will be stable. So ये है आपका इक्वेशन, फाइनल इक्वेशन जो राउथ टेस्ट फॉर स्टेबिलिटी क्राइटेरिया. So A0, A1, ultimately this characteristic equation that I told you, no, y 1 plus g equal to 0. 1 plus g equal to 0. G means what? The product of all the transfer functions. If you solve these equations, then ultimately you will get this equation like this. It's simply a polynomial equation. A0, A1, A2, A3, ultimately up to An is basically the coefficients of this. Right. Now look at if just 2-3 minutes I'm just finishing. If you see that A0, A1 all are positive. If the first value is negative, then you multiply by -1 and then you get the positive. But if you see the A0, A1, A2, A2, An all are positive, then system is stable. Understand that. You need not to do anything more. You do not know, just get the characteristic equation, and the characteristic, if you see all the coefficients, these are characteristic equation in Laplace domain. This S is there, that means Laplace domain. In characteristic equation in Laplace domain is there, and then all the coefficients are positive, then understand the system is stable. There is no danger of instability at all. But if it does not happen so, then they have to do this testing criteria. Here is the Routh criteria. You see that this characteristic equation already I have taken A0, A2, then A4, and all these things is given in the first line. Yeah, A0, A2, A4 in the first line, row 1. Then the other one, A1, A3. Look at the even and odd one. Even one, A0, A2, A4 are in the first row. Second row, that's the odd one, A1, A3, and all these things. Okay, A1, A3, A5, and all these things. And then the third row onwards, you have to generate. How to generate that I have shown you here. So B1 is nothing but A1, A2 minus A0, A3, and that then B2 will be equal to nothing but A1 into A4 minus A0 into A5, and that you do by A1. So this way the B1, B2, B3, this third row is created. C1, that means fourth row will be created like that, B1 into A3 minus A1 into B2 divided by B1. So that way you create this fourth row and all these things. And now all the criteria ultimately, if you this table if you complete, and you see all the values in the first row, first column are positive, then say that system is stable. आपको bother नहीं करना है. But if there is a change in the sign, right? Say 2 values are positive, 3 values are positive, and next is there is a minus. So that means how many roots will be there on the other side? That means as many sign changes are there. That means plus to minus, minus to plus. That means at least two roots will be there on the right side of the organ plane. Right. So this way you can find out the criteria for stability. So for the bioreactors and all this thing, mostly if you are really going to the industries and all this thing, bioreactors you have to run, and then if you have to find out the stability criteria and all these things, then you can understand that where you are operating, particularly in continuous mounted, the system will be stable or not. ये तुमको ये करना है. आज मैंने I not go details much into this. एक बोल के छोड़ देता हूँ. Now look at this. I have just given an example. I have given. This is the characteristic equation is to the power 4 TSQ and all this. These are 4-degree polynomials are there, and I have now look at all the coefficients are positive. So you don't know. So this system will be stable. And then Routh criteria are developed, and so you look at 1, 3, 11, 3, and all these things. And the first column is positive, so it will be stable. Now, thing is that now when the system will be now after the controller change is there. Now you want to see that controller working at the optimal level. ये भी तो देखना है, because then only system will be most profiting one. So then I will look at as again pole 0 baat are there. Then the denominator whatever things P1, P2 are the poles, and Z1, Z2 are the zeros. Pole 0 we call it. So pole 0, and then if you look at here, I have given an example here. This is the final example. You see that how many roots will be there? This is a 4-degree polynomial. Naturally, four roots will be there. So these 4 roots look at R1, R2, R3, R4, and where they will travel when the changes in the controller value. Controller, this is proportional controller I have taken, only not PI controller and PID controller, only proportional controller I have taken, and the K value is changing. Now 0, 0.23, 0.29, and all these things you change, corresponding changes and all this, what will happen to this? Now look at This is all minus, minus, minus. So that means throughout the left hand plane only will be there. The R1, the first root will be always in the left side of the organ plane. R2 will see, look at R2 second root, all are minus. Look at for different changes of k value. You see up to infinity. From 0 to infinity, I have changed the k value. And you see the R2 will be happen minus to minus 2. So full all are minus value. Sorry, right? Say on in this equation if you put. Because this k value, they just put this k value in this equation, this S4, S1 only, k value, you put it these values, you will see that all the values are this. So all are negative. So again, R1 and R2 both will be in the left hand plane, left hand side of the organ plane. Then R3 and R4 you look at. Here for the problem is there. So R3 is there, you see, up to this 3.384 k value, it is giving negative. But the moment it is changing to just small changes, 3.38 to 3.84, then you see the positive. That means going to the right hand side of the organ plane. Similar things as for R4 also. That means that now system can be unstable. Can be a system which is looking like stable, but thing is it may be stable, unstable if the controller set point, controller value, the k value, if you change, go beyond 3.384 or something like that. If you change beyond that, then automatically things will be unstable. तो आपको या तो सिस्टम को ऑपरेट करना है at the optimum point, and the optimum point when it will be for the maximum profitability. Basically, all your product will be best quality things, and you will get them at a minimum time. So if you want to get that, then you have to controller also set to the optimum value. And the optimum value, and if you, okay, you want to give more value than also 26, 28 and all these things, then the controller will be unstable. Not controller will be unstable, system will be unstable. Ultimately, system will be going beyond control. And so look at, these are the stars, four roots are there. Now how the four roots loops are there. So first two are no problem, right? But second, third, and all this thing that is changed. Look at this one is going to this one. The fourth one is going to this, and then going to this, and then this changes. So look at this I have given R1, R2, R3, R4. How changes? R1 goes from -3 to 2.69, then minus 2.69 to 4.6, and then minus 4.69 to 0.9. So R1 is totally a minus side में रहता है. R1 plane के minus side में. That means in left hand side. But then R2 also remains the totally full. Look at up to from anything up to infinity. If you change, then you will say R2 is negative values, no problem. But R3 you should look at. R3 goes from -1 to -1. यहाँ पर stable है. Then but then 0 to infinity. यहाँ पर से unstable हो गया. यहाँ पर से. So this is, look at, this is the value. It's going here. This is right hand side chala jayega. This is the locus. This is called locus of the root locus is, theory. So this root is going to the right hand side of this. That means controller को आपको design करना है such a way. Controller design is important. So such a way that you get the best output. Now R4 is also like that. So R4 का आप देखिए यहाँ से गया, this is going to this side. Look at R4 is going this side. So that means, though same system may be stable and may be unstable depending on how you choose the values, right? That's the root locus will tell you. Okay, so here I'm saying finishing. Anyway, some of the students, this is my research labs. Any question is there? Hello.
Any questions? We have due to a time crunch, we have a very short time, so only one question can be taken. So do you have any questions? So there is one question. In process chemistry, from Namrata Ray, in process chemistry, while scaling up from laboratory to large-scale production at primary manufacturing sites, problems like oxidation or decomposition impurities crop up. How do you suggest such problems to be handled?
This is out of context of this one, this lecture particularly. Here I have talked about the controller and the reactor. The ultimately production has to be done in the reactor only. Any production has to be done in big reactors, right? So even insulin is also being produced in big reactors, right? In the biocon. So here I've talked about the reactor instability, where the reactor can be optimally run and where reactor can go stable or where reactor will be unstable. So that means, yeah. But thing is that the question that is there is altogether out of this area. Can you repeat the
Okay.
question?
Uh, Sir, in process chemistry, while scaling up from laboratory to large-scale production at primary manufacturing sites, problems like oxidation or decomposition impurities crop up. How do you suggest such
Oxidation. If
problems?
you see, how, it depends on how you are doing the oxidation. That means air supply and all these things. You have to look at the control loop. You have to check that whether control loop how much air is required and where the system will be. वो तो देखने से पता चलेगा जो कैसा system भी है. There are oxidation system is there in the reactor. If you have the real oxygen probe is there, and they measure the oxygen value in the liquid system, and all other ways. If you want to measure the partial pressure of oxygen, that also you can measure. But then you have to see that you have to look at the reaction requirement. How much reaction oxygen is required for the conversion, whatever conversion is doing, right? If it's the oxidative process and all these things. So that is mathematically can be calculated, and accordingly you have to supply.
Thank you, sir, for your insightful speech. So, and I would request the participants, because of the time crunch, please to ask no more questions. And whatever questions you all have, you all can send it to us so we could forward it to sir, and you will get your replies. And I just want to say another thing that please, I know it's a very short time, but please join us at 2 p.m. because the other speaker will join at 2 p.m. sharp, and the assistant controller would like to tell a few words. So please stay tuned for some time. Okay, she'll be telling it afterwards. So thank you, sir, once again. And we just now conclude this session for lunch break for 15 minutes, and come join again at sharp at 2. Thank you.
Okay, thank you. So shall I log off? Log off.
Yes, sir. Thank you, sir. Thank you very much.